Good morning, everyone. Welcome to the national webinar on diversified fungi, insights and implications. Fungi can be many things in our lives. They can be nutritious on our plate as plum buttons or be an itch and annoyance on the skin. They can help a huge tree flourish or cause a heavy loss to our crops. They can enter our vital organs and cause death or heal us with their antibiotics. They add flavor to our cheese and also rot our foods. They appear out of nowhere and disappear without a trace and yet spread their carpet for miles under the surface. Fungi truly are microbial marvels. Today we have amongst us scientists, professors and teachers who have spent better part of their career researching and working with these fungi. Let us appreciate these wondrous God's creation with our speakers today. We begin our webinar on an auspicious note with a prayer.
तनयमूर्ति शरणु मूषिकवाहना शरणु सिद्धिविनायका शिवहालम्बुधरणे इक्षुशापगगिलिदने शिवहालम्बुधरणे इक्षुशापगगिलिदने पक्षिवाहन श्री पुरन्दर विठलन निजधारणि पक्षिवाहन श्री पुरन्दर विठलन निजधारणि शरणु सिद्धिविनायका शरणु वेद्यप्रदायका शरणु पार्वती तनयमूर्ति शरणु मूषिकवाहना शरणु सिद्धिविनायका.
Thank you. We have with us Dr. R. Manimegalai, our principal, Dr. MGR Janaki, College of Arts and Science for Women. Ma'am, I kindly request you to deliver the welcome address.
Good morning. It's my pleasure to welcome Dr. Thangam Menon, Emeritus Professor, Department of Microbiology, Savitha Dental College and Hospitals, Chennai, who has joined us today to inaugurate the national webinar on diversified fungi, insights and implications, organized by the Department of Microbiology of our college. I welcome our correspondent, Dr. Lata Rajendran, Chairman, Dr. Kumar Rajendran, faculty members and students. Biodiversity is one of the most interesting aspects of biology. Fungi is an important component of biodiversity, which plays a major role in various ecological cycles. Fungi presents enormous species diversity with respect to morphological, ecological, and nutritional modes. The new assessment of fungal species calls for more knowledge on fungal inhibiting environments and the use of new molecular approaches. Therefore, cooperation and communication of all mycologists all over the world is crucial to study the fungal diversity. I'm sure this webinar will enhance the horizon of knowledge. I would like to thank and welcome all the guest speakers who will be joining us today and sharing their knowledge in this webinar. I would like to thank Dr. Lakshmi Priya and the faculty members of the Department of Microbiology for planning and organizing this seminar. I welcome you all once again. Thank you.
Thank you, ma'am. I now call upon Dr. Nitya Kalyani B., Head of the UG Department of Microbiology, Dr. MGR Janaki College of Arts and Science, to render the dynamics of the seminar.
A very good morning to everyone. Respected Dr. Lata Rajendran, Correspondent, Dr. Janaki College of Arts and Science for Women, Chairman Dr. Kumar Rajendran, Principal Dr. Manimegle, distinguished speakers, academicians, faculty members, participants, and my dear students. On behalf of the Department of Microbiology, I take immense pleasure in welcoming you to the national webinar on the theme Diversified Fungi, Insights and Implications, organized by the Department of Microbiology, Dr. Janaki College of Arts and Science for Women. Our planet is endowed with diverse groups of organisms. Fungi, the so-called fifth kingdom, plays a significant role in interacting with plants, humans, and animals, leading to symbiotic, mutualistic, saprophytic, or pathogenic life. Fungi play a very important role in addressing major global challenges, being instrumental for improved resource efficiency, making renewable substitutes for products from fossil resources, counteracting lifestyle diseases and antibiotic resistance through strengthening the gut biota, and functioning as host organisms for production of new biological drugs. This range of the potentiality of fungi all stand on the shoulders of the efforts of mycologists over generations. The scientific discipline mycology has built comprehensive understanding within fungal biodiversity, classification, evolution, genetics, physiology, ecology, pathogenesis, and nutrition. To unfold the full potentials of what fungi can do for both environment and man, this national webinar is themed on offering a scientific framework to make the scientific community be aware of the immense importance of fungi and for expanding the exploitation of fungal biodiversity for more value-added uses. We appreciate greatly and heartily welcome our distinguished guest, Dr. Thangam Menon, Emeritus Professor, Department of Microbiology, Savita Dental College and Hospitals, Chennai, who has graciously accepted our invite to deliver the inaugural address on fungal infections in the COVID era, and our esteemed speakers, Dr. Bernard Roderick, who is a senior professor in botany discipline at the School of Biological and Biotechnology, Goa University, Dr. Prabhu, assistant professor and head in charge, the Department of Microbiology at Dr. ALM PGIBMS, University of Madras, and Dr. D. Aravind Prashant, associate professor in Department of Microbiology at School of Biosciences, Periyar University, who are all here to impart their knowledge on various issues relevant to the theme of the webinar. I once again thank our esteemed speakers for accepting our invitation and would like to extend my gratitude towards our management and our principal for encouraging us to organize today's national webinar. I hope you all find this webinar engaging and feel enriched with knowledge on completion of this event. I once again welcome you all to the webinar and thank you all for the participation.
Thank you, ma'am. We have with us Dr. C. Bhanureka, Assistant Professor, Department of Microbiology. She will be introducing us our guest for today, Dr. Thangam Menon.
Professor Emeritus, Savita Dental College and Hospital, Savita University, inaugural address for a national webinar on diversified fungi, insights and implications. Dr. Thangam Menon is a retired professor at Department of Microbiology, Dr. ALM PG IBMS, University of Madras, Chennai. She served as a senior demonstrator, Department of Microbiology, CMC Vellore, and as research fellow, lecturer, reader, professor, and ed in the Department of Microbiology, Dr. ALM PG IBMS, University of Madras from 1980 to 2017, and as UGC BSR faculty fellow, University of Madras. To add on, Madam was member syndicate University of Madras from February 2013 to 16. As director, Dr. ALM PG IBMS, University of Madras from November 2012 to 2014. As chairperson, School of Basic Medical Sciences, University of Madras from July 2015 to 2016. Madam's research specialization is in infection in immunocompromised patient, streptococcal infections, infective endocarditis, and oral microbiome. Madam has more than 40 years of research and teaching experience. She has guided more than 31 PhD students and 91 PG students. She has more than 183 research papers in national and international journals. She also published a book on novel coronavirus, SARS-CoV-2, and overview in May 2022. To her credit, she has many ICMR, UGC, DBT, DOD funded research projects. Madam is a expert committee as UGC SAP review committee member, UGC expert committee member for microbiology, UGC expert committee for award of Raman Fellowship, Government of India Central Drug Standard Control Organization, Biotech Park for Women Chennai as advisory committee member, expert member of Technology Development Board in Department of Science and Technology. She is a proud recipient of many awards. To name a few, British Council Awardee in 1992, Siri Research Award in 1992, Agathir Award in 1999, Tansa Award in 2006, ICMR Senior Scientist Awardee by University of Texas, USA in 2008, Academic Achievement Award in 2012, Lifetime Achievement Award in 2014, Professor Sundaram Subramaniam Oration Award in 2017. She is a member of Board of Examiner in University of Madras, Anna University, Dr. MGR Medical University, University of Kerala, University of Mangalore, Bangalore University, Karpagam University, Pondicherry University, Calicut University, Mysore University, and in Savita University. She is a Board of Studies member in Pondicherry University, Mysore University, Tiruvallur University, MGR Medical University, Goa University, and in Central University of Tamil Nadu. Patent was awarded for an improved medium for probiotic assay and a process for its preparation. Madam, you keep setting the bar higher and higher, and we are amazed by the passion you show to your work. Waiting for your words of wisdom. Thank you.
Good morning. I'm audible.
Yes, ma'am, you are audible,
Respected,
ma'am.
respected members of the management committee in Janaki College, the principal, Dr. Manimekale, and the members of the microbiology department, Dr. Nitya Kalyani, Dr. Lakshmi Priya, and others. Thank you for inviting me for this webinar. It gives me great pleasure to give you the inaugural address. I first would like to congratulate the microbiology team for having chosen such a relevant topic because, fungi, as we all know, are microorganisms which have touched our lives in so many ways. And at the same time, today we live in the COVID era. So I thought I could, in line with the theme of your webinar, I thought I could, give you a short, brief lecture on the way the fungal infections have impacted COVID patients and, whatever information that we have today on the fungal infections in patients who are very ill with COVID. So now this link that we have of COVID and fungal infections, we know that those who have got very severe COVID infections are at high risk of fungal infections. We see this particularly in patients in the ICU and so on. And there have been many reports of COVID-19 co-infections by many fungal pathogens, especially in critically ill patients. Now, this happens because there is COVID brings about a lot of damage to the lungs, particularly the epithelial cells. It brings about decreased blood counts, and generally it brings about a dysfunction of our immune system. To add to this, we have a problem that these patients are often treated with antibiotics and with steroids and also with some immunosuppressive agents, all of which have an effect on the neutrophils and the macrophages and bring down our immune response. Some of these patients also have classical predisposing factors like diabetes and renal failure, and some of them may also be on central venous catheters, and all this adds to this problem. uh, the three kinds of infections, fungal infections, which are classically associated with COVID are candidiasis, aspergillosis, and mucormycosis. So, because of the increase and prevalence of these, particularly these fungi in COVID, we now have new terms for this, which we never had before COVID came into existence. So, we call this COVID-associated candida is known as CAC. COVID-associated pulmonary aspergillosis is known as kappa, and we have mucormycosis, which is CAM, that is COVID associated mucormycosis. So these have now become specific disease entities, and we never used to use these terms before COVID. So coming to candidiasis, COVID associated candidiasis can either be superficial candida infections of the skin, or they can be invasive infections, and their prevalence ranges from about 1% to about 25%. Among these, Candida albicans is actually the most invasive of the lot, but you do get infections with other species like Candida glabrata, Candida parasilosis, Candida tropicalis, and very recently, Candida auris, which is a new species, an emergent pathogen, and something which we have all been very concerned about. So, this is considered a new emergent global threat for several reasons. Most important being that it can sometimes be resistant to antifungals that we normally use, like fluconazole and amphotericin B. And we mostly they probably would be sensitive to a group of antifungals which we call echinocandins, but echinocandins are not readily available in all countries, so that again creates a problem. We also worry because Candida auris is very quickly transmitted, and it is presumed that sometimes this is possible because the transmission can happen through healthcare workers, because they do not use the PPE, the personal protective equipment, correctly, or they use it for too long, which means it can be contaminated, and this can lead to cross contamination and transmission. No, Candida auris was first isolated um, in Japan 2019 from the external auditory canal of a patient. And since then, there's been a global emergence of this fungus across all continents except Antarctica. Now, currently, this is separated into four clades, and each clade is different by more than 10,000 SNPs. So we have the South Asian clade, the East Asian clade, the African clade, the South American clade, and we also have now a fifth clade, which is a clade from Iran. Because there is a lot of variation, SNP variations between these different clades, this suggests that Candida auris actually comes from multiple locations, and it is not a single clonal source because it is simultaneously coming from different locations at different times. So it's not probably coming from one clonal source. And genetic analysis of these Candida auris has shown that it's very different from all the other Candida, but it is closely related to one or two species, one important being the Candida hemoluni and Candida lusitanae. In India, also studies have been done, and we have seen this in many parts of India. It has been reported, but within each geographic region within the clade, there are not so many SNP differences. So, Candida auris has been associated with outbreaks in intensive care settings, and this has been we have had a lot of transmission in spite of having good infection control policies. And as I said before, this is probably happening because the healthcare workers are transmitting the infection, probably with contaminated PPEs. And when patients are very ill, the infection spreads very rapidly because their immune system is also low. And another problem is that the antifungal susceptibility profile of auris can never be predicted. It is very variable, and sometimes some strains are very resistant. And also, they can become resistant after they are exposed to antifungals. We also have a problem in identification using conventional phenotypic and molecular techniques. We don't know the actual prevalence, and we don't know where in the environment the organism is present. So these problems have made it difficult for us to actually control the infection. Auris is related to the Hemolini species, and Hemolini, we know, is associated with deep-seated soft tissue and bone infections, particularly in immunocompromised patients, particularly diabetes. We see that Hemolini is less frequently isolated than Auris, but it is probable that many species of Hemolini are actually misidentified as Candida auris. We do use a chromogenic medium to differentiate between auris and Hemolini isolates. And so this chromagar is probably a low-cost method which can be used in place of phenotypic assays. Now, Candida auris is actually misidentified in many ways. If you use normal phenotypic tests, the traditional methods like VTEC2, API, etc., we find that this Candida auris can be misidentified. So the best method of detection is actually by Malditoff. And Malditoff is now a method which we use for detecting or for identifying many microorganisms. And it is based on the fact that when you add a sample to a matrix, an organic matrix, the matrix embeds this particular sample, and when you use a laser beam, this gets ionized, and these protonated ions actually travel through a field, and they can be detected on an analyzer. So mostly we use the time of flight analyzer. That means the time taken for these ions to travel within the flight tube. And the problem with MALDI is that it actually compares the spectra which you find between each sample. So they are detected based on the spectra. So the problem is that your database must have all the spectra. So if you're using MALDI but you don't have the orange database, then you will not detect it. It will probably detect it as something else, Hemolini or something else which is related to that. So when MALDI is used, it's very important that you need to have the database that will actually detect it in the sample. Now, there are also other methods for detecting Candida auris. One is the AFLP, which is the amplified fragment length polymorphism, very similar to RFLP, except that this AFLP requires that you amplify the digested DNA. Then you have pulse field gel electrophoresis. Then you have the M13 DNA fingerprinting, where you use a bacteriophage M13 and sequence the genetic loci and hybridize it and then sequence the genetic loci. And then we have the methods of 28 srDNA or the ITS, which are both frequently used for taxonomy in fungi. D1, D2 domains are the regions near the F dash region of the gene, and these are very variable and they are specific. So we use this to identify the fungi. Now, coming to the growth of Candida auris, it grows at 40 to 42 degrees on most medium. It can also grow on chromogenic medium, where you get whitish or pinkish or purplish colonies. Again, not very characteristic, because those of you who have used Chrome Ega will know that these white, purple, beige, and mauve color you can get for many Candida species. But this is what it looks like, and it doesn't grow in the presence of cyclohexamide. It does not form germ tubes, and microscopically they are round or oval, more often oval, and you normally never find pseudohyphae like you see for other Candida species. And when you look at the profiles of the assimilation profiles, what is typically seen is that N-acetyl glucosamine has been found to be assimilated in the isolates from India, but this is different from other countries. Those which have been isolated from Japan, South Korea don't assimilate N-acetyl glucosamine. So we know that in certain other cases of Candida, we identify based on the assimilation profiles. That is a little difficult here because they don't have very standard assimilation profiles. They also produce extracellular enzymes like proteinase and phospholipase, and they do form biofilms. Um, the treatment of these infections, many of them show high MICs to fluconazole. Of course, this is not there in every strain, but you should expect it that when the auris is isolated, it is probably resistant to fluconazole. It can also be less sensitive to other triazole antifungals, including things like voriconazole, itraconazole, which are very frequently used. And coming to amphotericin B, also you can get a lot of variation in the susceptibility. So when you want to have an empirical treatment for Candida auris infection, it is better to give the patient echinocandin. But echinocandin is also not very easily available all the time. And among them, micafungin has got the best efficacy. And but micafungin is now so often used that we still worry that we might have resistance to micafungin also. There have been reports of auris from all parts of the world, and this is one small report which was reported in a Mexican hospital where they isolated 15 strains, of which 12 were from patients and 3 were from the environment. But they found that all 15 were resistant to amphotericin B and fluconazole, and most of the patients died in spite of antifungal treatment. And so this is in a place where there was very strict control of risk factors, and there was antibiotic stewardship, and they also were careful in the use of steroids and so on, but still they found that there was a high mortality rate. Now, there have been studies in India, and in 2009 to 11, 12 isolates were identified in 2 hospitals. This was reported. All the strains were resistant to fluconazole, and they were genotypically distinct from the strains which were recently isolated from Korea and Japan. And this they have identified by doing M13 fingerprinting and AFLP. And all these strains were oval forms, and they were negative for pseudohyphae and negative for germ tube formation. They grew between 37 and 42 degrees, and actually all of them were misidentified using the classical systems that we use. We take 2 misidentified 10 of them, mostly as hemolini, and the API system misidentified them as Candida saccade. And all of them assimilated N-acetyl glucosamine. They were identified by the ITS D1, D2 regions, showed 100% homology to database strain of C. auris isolate, and 98% similar to the neighboring countries what we isolated. Um, and they also showed up to 98% similarity with the closest hemolinic complex. So you can see how difficult it is to identify Candida auris even at the molecular level. So the next organism we would be talking about is kappa, or Candida associated pulmonary aspergillosis. Now, this was we presume that there's in patients who have got severe COVID-19 infections, there's an estimated prevalence of at least 5%, and it could be much higher in patients who have got acute respiratory disease syndrome, and they have high IL-6 levels. The one problem of identifying is that it does not have typical radiological features of aspergillosis, because when it happens in COVID-19 patients, the radiological pictures are slightly different. And another problem is that in invasive aspergillosis is more common in immunocompromised patients, whereas kappa is seen also in immunocompetent patients when they are suffering from ARDS and when they require steroids. So the pathogenesis of this infection is, it means that typically there is a very severe inflammatory response, and this leads to the sloughing of the epithelial cells in the airways. And when this happens, the conidia of the aspergillus are able to invade the airway, and this leads to inflammation and necrosis. Um, the viral infection actually hampers the ciliary clearance and leads to immune dysfunction or dysregulation, or both. There are actually three grades of the disease: possible, probable, and proven, for use in clinical delineation of patients, and to classify them for also for clinical trials. When Candida 19 associated aspergillosis is there in a patient, it can definitely worsen the disease course of COVID-19, and so it can increase mortality. The first line therapy is voriconazole or isavuconazole. And if there is an azole resistant, then amphotericin B can be used. Immune dysregulation or immune suppression is something which we see for frequently in COVID patients, and this is what facilitates the super infections with bacteria and fungi. We also see that there's a distinct decrease in the T cell populations in patients who are severely infected. And when you have a decreased lymphocyte count, this can be also accompanied by a defective function of the lymphocytes. So this is IPA is a very serious secondary infection in COVID patients. It has mortality up to 25% more than what it would be in the absence of aspergillosis. Now, we don't know very clearly whether the infection, SARS infection itself, is a risk factor, or whether additional factors like steroid therapy are risk factors. But we know that chronic steroid treatment, when it is given to patients, they are more frequently getting this kappa. And also the steroid use was more frequent in patients who died of these infections. So it is presumed that the dexamethasone, which is normally used, and the anti-IL-6 directed therapy may increase susceptibility to superb infections. And one of them is the invasive pulmonary aspergillosis. So, as I told you, this aspergillosis is usually seen in immunocompromised patients, whereas you can see kappa also in immunocompetent patients who are suffering from ARDS and who have been treated with steroids. And another thing is that other usual risk factors like old age, chronic lung disease, and diabetes, which is not controlled, antibiotic use may not always be present. Now, when we look at the radiology, this is a picture from a report from medical mycology case reports of a patient who had COVID-19 patient who had aspergillosis. So what we can see is multiple pulmonary nodules or lung cavitation. And so most of them have these ground glass opacities, which you can see in the A, B, and C. You can see that it is progressively, these consolidative patches are progressively increasing A, B, and C. And then after this, a patient is treated with voriconazole. So the last slide is a one which is taken after 1 week of voriconazole treatment. And you can see that there is overall improvement. Now it is also difficult to diagnose because many of these pictures of opacities, consolidation in the lungs is seen in many diseases. So so this also can be differently presented in the CT. It is normally what they try to diagnose is by doing a culture from a transtracheal aspirate, a fungal culture. If it is there, it is quite easy to grow in culture. And then we have this aspergillus galactomannan test, which is the galactomannan is a polysaccharide antigen on the surface of the cell, and that is tested for in it is positive in invasive aspergillosis. So when this is positive, it can be presumed that the patient has got aspergillosis. Uh, then you may not see the classical angioinvasive aspergillus findings like cavities, halos, etc. may not always be seen in COVID patients. And even the galactomannan test, it is not such a sensitive test in all cases. It is better when you do it from the bowel sample rather than from the serum sample. So maybe 70 to 80% of bowel samples may yield a positive, but less than 50% of the serum samples can give you a positive. So it is quite difficult to diagnose. Now we go to the third group of infections, which is the mucormycosis. These are caused by mucormycetes, these are fungi which live in the environment, and they also live in soil and decaying matter and in rotten wood. So you find it everywhere in the environment. Um, they are characterized by fungi which are aseptate, that is, they don't have a segmented thallus. And the reason why COVID-19 patients get mucormycosis, though it's still debated, it is presumed that is because of three main reasons. One is the immune dysregulation and the ciliary dysfunction that you see in COVID, and the severe inflammation. And second is the hyperglycemia, which will lead to the dysfunction of the polymorphs. And then the third is the corticosteroids, which actually decrease the immune response, impair neutrophil migration, and impair the phagolysosome fusion. And also, in many cases, it increases the hyperglycemia. So this is a three-pronged reason which is given for the reason why mucormycosis happens in these patients. So there are many fungi which can cause mucormycosis. Most commonly are the rhizopus. And there's, of course, mucor species, the rhizomucor, the syncyplastrum, Cunningham-Hella, Apophysomyces, and Listeria, which Listeria was earlier known as Apsidia. So what is very characteristic of mucormycosis is that you have tissue necrosis, and this is can be seen as a black discharge or an eschar in the nasal or the oral cavity. Patient may or may not be having fever, and most of the time, other features indicates the involvement of thrombosis. The different types are the rhino-orbitocerebral type, the pulmonary, the gastrointestinal, cutaneous, and the disseminated type, of which the the most important is the rhinocerebral type. There is a necrotic skin, black discharge. There can be ulcers. There can be facial swelling, visual impairment, sinus infection, and black discoloration in patches, and a non-remitting febrile illness. It is mostly diagnosed with an MRI and or with an endoscopy of the collection of tissue, that is a biopsy. Then this is stained, microscopy. They look for the non-septate hyphae, and biopsy is the most important way it is diagnosed. Um, the pulmonary type of mucormycosis presents as fever, cough, chest pain, hemoptysis, and generally worsening of the respiratory symptoms. On the chest X-ray or the CT, you can see the reverse halo sign. You can see many nodules showing pleural effusion, and the galactomannan test and beta glucan D test will normally be continuously negative. Now, this reverse halo sign is seen very often in many kinds of diseases. So it's not very typical for this. It means there's a small ring, and this is surrounded by a halo. So that's why it's called reverse halo. I think I had a slide, but I think the slides which are projected are not the slides which I've made now, but what I showed you yesterday. I think that was the problem. So I don't have many of the pictures which I actually made more recently. So that shows you a typical reverse halo sign. So if you can make this out, there is this is the reverse halo sign. This shows a small area of consolidation, and this opacity surrounding this, and that's why it's called a reverse halo. So this can be how it is treated is first by controlling other factors, particularly diabetes and diabetes-related factors, and being careful about the steroids. If the patient is still on steroids, you have to discontinue them rapidly. Also, discontinue all the other immunomodulating drugs, and very important to do surgical debridement, because unless the dead tissue is removed, it is very difficult for the infection to improve. And surgical debridement is a very important step, and it's important to do it well, extensively, to remove all necrotic material. And it's very sad if the eye is involved. It frequently requires exentration of the eye. It is managed with amphotericin B, different forms, ordinary amphotericin B, liposomal, lipid complex, etc. Then with adequate hydration, and by monitoring the renal function, particularly the urea creatinine, and also the serum potassium. Some patients who are not tolerant to amphotericin B can be given posaconazole. And given this, it's important to being careful not to expose to decaying organic matter in the environment when you know that it is likely to be contaminated with these organisms. Controlling hyperglycemia, and particularly monitoring the patients who require steroid therapy. Important that steroid therapy is given in the right time, the right dose, and otherwise it can really lead to problems. And many people who treat COVID-19 patients know this. So antifungals and antibacterials should be used only when they are needed. So I think this is the last slide. This is just to tell you that this era of COVID-19 has taught us many things. One is that the COVID-19 and fungal infection, co-infections, can be very serious, and so we need to find more ways to understand the pathogenesis of these infections. We need better antifungal agents. We need good diagnostic modalities and better management policies. And it must be very important for everybody who treats these infections to understand the possible complications in order to decrease morbidity and mortality in these patients. Thank you very much.
Thank you, ma'am. Ma'am, we would like you to address few questions from our participants, ma'am.
Yeah.
May I read it out?
Yes.
The first question is, how can we boost our immune system to avoid these super infections?
See, there are many ways of boosting the immune system, which we do as home remedies. There are many things that we do. All of us know that we've been doing all these things, ginger, turmeric, and all these kind of things. Many things which can improve. But on the therapeutic point of view, it is very difficult. What they do is they normally try to see whether you can give a prophylaxis. If you know that a patient is most likely candidate to develop this, you can give a prophylaxis. But this needs a lot more study, because prophylaxis of giving an antifungal is not as simple as giving an antibiotic antibacterial, because antifungal itself has a lot of effects on its own. So it's very difficult to give a prophylaxis, but this is still being thought about, whether a patient who is more prone to this, you can give prophylaxis. Otherwise, it's there are some routine things. You can give some multivitamins, like vitamin C is known to be has many people have found that vitamin C, zinc, things like that do increase. And I think many of us have been continuously taking vitamin C and zinc. We don't know how effective it is, whether it's belief, or of course it has some scientific reason also. So I think that's one way you can increase your immune response. Eat well, eat healthy, and exercise, and live a more disciplined life in all respects. Probably your immunity would improve with that.
Yes,
But it's
ma'am.
very difficult to have a medicine for to stay straight away. You can improve unless you give a straight an antibody where you want to actually target a particular organism. There's no other way of doing it except that. And there are immunomodulating agents which will actually improve, but then they have many kinds of effects. So whether you can give it on a regular, whether immunomodulating agents can be given on a regular basis, we still don't know. May not be very appropriate.
Okay, ma'am. We have another question.
Yeah.
Does temperature and or weather pattern have any impact on the spectrum of the fungal diseases?
Yeah, because weather, temperature, because humid conditions we know will always favor. Always humid conditions favors fungal growth. And so you remember that there was a time when everybody did steam inhalation for COVID. Everybody did that. Then some reports were there that could steam be one of the reasons by which people are getting more COVID. So then people decrease that. They don't do it so often. They are worried whether that will bring about mucormycosis and so on. But generally, humid conditions are known to promote fungal infections, and weather conditions. This weather which is cold and has a lot of the atmosphere is quite laden with many of the fungal spores. So that can again, if you inhale that, many people who go out in the cold weather are more prone to getting these infections than otherwise. So warm weather is a little better when you look at it. Cold, humid, all this brings about humidity is good for fungal growth.
We have just a couple of questions more, ma'am. What is your view on common fungus as threat to children in post-COVID era? Views on common fungus as threat to children in post-COVID era.
That is, see, in children, what kind of fungus infections you see are mostly these dermatophytes. Most children have only that. They don't come unless they are having some other illness. A normal child would not have any other fungal infection. See, one thing what we need to understand about fungal infections is that mostly, unlike bacterial infections, if you are close to normal, you will not get a fungal infection. So in children, the only kind of fungal infections which are commonly seen are the dermatophytes, which are the what we commonly called ringworm infections, skin infections. So that has nothing so much to do with the post-COVID era, though some people have reported more skin allergies after COVID. I don't think there's any increase in dermatophyte infections after COVID.
Okay, ma'am. One more question. That will be the last question, ma'am. What are the biomarkers available for the diagnosis of invasive aspergillosis?
See, you have some tests, like I explained to you, the galactomannan test, which was once considered a very good test, but it is a polysaccharide which is on the cell wall and which is goes into the patient, the host, when during an acute infection. But now it is, everybody doesn't go so much by that, because galactomannan, if you do it, you must do it from a bowel sample, and you should. If you do it from a serum sample, you may not get such a good result. So, as I told you, there were studies which show that serum samples give less than 50%. So, bowel samples are not very easy to collect, and especially COVID, it's extremely difficult to collect. Actually, if you want to invasive aspergillosis, the best way to diagnose would be by a biopsy, which shows infection, and by a culture. The culture is typically done 3 times consecutively, and if you get a aspergillus in one culture, we don't go by that. You need to take it consecutively on 3 days and see whether every day for 3 times you isolate aspergillus. In that case, and there is a reason, the doctor may do a biopsy. Otherwise, if he believes the laboratory is doing standard this thing, they probably will just treat with an antifungal with that. So that is much better. Aspergillus is very easy to grow. So we don't necessarily use other biomarkers routinely, because a culture result would be more, more confirmatory.
Thank you, ma'am. Thank you very much for enlightening us with the impact of fungal infections in COVID patients in relevance to the current scenario. It was an honor to have a highly experienced person like you in the field of mycology amongst us. Once again, thank you very much, ma'am.
Thank you so much. Thank you for having me.
We would now have a photo session. So all of us would be included in the screen, and it would be an online photo session.
Okay.
Is it done, ma'am?
Yeah.
Thank you, ma'am. Thank you so much for enlightening us on fungal associated COVID diseases, ma'am. Thank you for your time, ma'am.
Thank you so much.
Thank you so much, ma'am.
Thank you. Bye-bye.
Thank you, ma'am.
Thank you all. We would now begin our sessions. I now request Dr. Vidya Lakshmi, Assistant Professor, Department of Microbiology, to introduce our first speaker.
A very good morning to one and all gathered here for our webinar. It gives an immense pleasure for me to introduce a renowned speaker, Dr. Bernard Rodriguez, who is a senior professor in botany discipline, School of Biological Sciences and Biotechnology, Goa University. Dr. Bernard has over 3 decades of teaching and research experience. His research interest revolves around mycorrhizal fungi that includes identification of novel AM species, AM fungal diversity in various habitats, taxonomy, inoculum production, screening studies to identify efficient AM species for various crop plants, plantation crops, and ornamental crops. His area of other interests include endophytes, medicinal plants, and remote systems. Dr. Rodriguez has an excellent academic record. He had done a dozen of major and minor research projects funded by various state and national funding agencies. Dr. Rodriguez had attended over 165 national and international conferences, workshops, seminars, delivered plenary and lead lectures, and has also chaired several scientific sessions. He had published 151 research papers in national and international journals of repute and edited 5 books. He had guided more than 14 research. He is also editorial board member of several journals, reviewer for a number of national and international journals. Dr. Rodriguez is lifetime member of many prestigious bodies: Botanical Society of Goa, Mycological Society of India, Indian Society of Mycology and Plant Pathology, Indian Mycological Society, Indian Society of Plant Pathology. Dr. Bernard has won multiple accolades. Twice the Young Scientist Award from DSC New Delhi, Excellence Award from Tanzanian Parents Network Foundation, Professor Bide Memorial Award by Indian Society of Mycology and Parasitology, Best University Teacher State Award from Government of Goa, Dr. Agnihotradu Memorial Award by Mycological Society of India. It gives an immense pleasure for us, sir, to invite you to share your great knowledge and propel this event forward. Thank you.
Sir?
Yeah, just a
Yes, sir.
minute.
You can begin with your presentation.
Yeah, good morning, everyone. Am I audible?
Yes, sir, you are audible, sir.
Fine. Yeah. Well, at the outset, let me thank Dr. Vidyalakshmi for a nice introduction. Uh, Principal of Dr. MGR Janaki College for Women, Dr. R. Manimikalai. Then the head of the UG Department of Microbiology, Dr. V. Nitya Kalyani. Faculty members uh, MGR Janaki College for Women, uh, delegates and student friends. At the outset, let me congratulate the Department of Microbiology of Dr. MGR Janaki College of Arts and Science for Women for taking the initiative to organize this national webinar on diversified fungi, insights and implications. I thank the organizers for giving me this privilege to participate in this webinar. I'll be talking on the topic mycorrhizal fungi, a topic that is very dear to my heart. And I've been working for quite some time trying to understand the, the different roles of mycorrhizal fungi. And I'll be talking on the topic, as I said, mycorrhizal fungi. Now, before that, we know that agriculture is very important. Agriculture started some 10,000 years back, where man has been cultivating crops like rice and wheat and so on. And therefore, it is essential for our survival. And then we need to look for ecologically friendly agricultural techniques which can, in a way, provide food to us. At the same time, it can nourish the ecosystems. And when we talk about uh, agriculturally, ecologically friendly agricultural techniques, then we also have to know that our ancestors have been using the organic manures like farmyard manure, cow dung and different other manures, and then later on we switched on to inorganic fertilizers, which has created a problem, and which has made the soils acidic and inert, so that on such soils agriculture is not possible, and there are large tracts of land which have been converted into wastelands. And today we are looking at sustainable agriculture, the technique that would safeguard our ecosystem, our environment, and the human health. Here, when we talk about these microorganisms and when we talk about the plant, then the soil is important, because most of the time we look at the above ground portion of the plant and nobody bothers about the portion that is below ground. And the soil is important because the soil has variety of beneficial microorganisms. And it is said a country that has rich soil is a rich country. So therefore, these microorganisms have several roles to play, several benefits you can provide to the plants, uh, and in terms of fertility, I mean in terms of nutrients, and that makes the increase the productivity of the plant. So these organisms, we need to sustain them in the soil so that they can multiply through our agricultural practices, through eco-friendly agricultural techniques. And to now to go about from inorganic to organic or sustainable agriculture, then we have to study the microbes in detail, their characteristics, their effectiveness, consistency, and precautions, limitations have to be studied not only at the laboratory level but also at the field level. So, with this in mind, we talk about sustainable agriculture. Today, I'll be talking on the topic the mycorrhizal fungi. Yeah, before that, the different type of microbial inoculants which are used. So we have the rhizobium, azotobacter, then the azospirillum, then blue-green algae, then and then EM fungi. So different organisms which are involved. So I will be talking on the EM fungi. Now coming to the mycorrhizal fungi, they are the colonists of plants and soil. So you can see here the how the plants they grow in the ecosystem, and the rhizosphere, the root are associated with these mycorrhizal fungi. So there is a web of these mycorrhizal fungi in the soil, and this web is so huge that it is spread over the entire globe. You find mycorrhizae in almost all ecosystems, may it be arid or it may be, I mean, the deserts and the even in the hostile environments. So we have the plants, the soil, and the mycorrhiza. These are the three components. Now, why is mycorrhiza important? Because they help in the uptake of nutrients. It's a symbiotic association where both the plant as well as the fungus is benefited. Now, how the plant is benefited? Because the the plant uh, gets nutrients, especially phosphorus, whereas the fungus gets carbon compounds. Plant being autotrophic, it can produce its own food through photosynthesis, whereas the fungus being heterotrophic has to depend on the plant for its carbon compounds. Therefore it is a give and take relationship where the plant provides about 20% of the photosynthesis to the fungus. Now when it coming to the how the what is the main role of these EM fungi. Now when these EM fungi they are associated with the roots, you can see here the hyphae are associated with the roots, whereas here you find that only the roots. So the roots are able to absorb nutrients only from the limited area, whereas when they are associated with the mycorrhizal fungi, then these fungi can go far and beyond in the soil where the roots cannot reach and can provide nutrients. Then uptake of mainly uptake of phosphorus, which is very very important. Now phosphorus is a very sluggish element, and then in the soil you find that this is the root, and this root uh, can absorb within that is available within the vicinity. So these phosphate ions which are there, they are not available because the roots have absorbed what is already in their vicinity. So for this phosphorus to move from the uh, to the root zone, it will take much time since phosphorus is a very slow moving nutrient. But when it is associated with mycorrhizae, you find that these phosphorus ions, the mycorrhizal hyphae fungi can take up, and then they can, through the hyphae, it can be transferred into the root system, thereby helping better plant growth. Secondly, the tropical soils are deficient in phosphorus. Tropical soils being deficient in phosphorus, therefore mycorrhizae play a great role in the phosphorus uptake and therefore the plant growth. Now, when you talk about the deficient in phosphorus means when you add phosphatic fertilizers, inorganic fertilizers, you find that as far as the phosphatic fertilizers are concerned, 85% of the phosphorus gets fixed in the soil, whereas only 15% is available to the plant. So lot of it is lost or is wasted. Now how it is wasted? Because this phosphorus that is added to the soil, it would form, it would combine with like calcium in the alkaline soils, whereas it will combine with iron and aluminum in the acidic soils. So when it forms a bonds with phosphorus and with either with iron or calcium or aluminum, then it is in the bound form and is not available. Therefore, the mycorrhiza would play a role. Even again in the soil, there are PSBs or PSMs, that is phosphate solubilizing organisms or bacteria, which have the ability to produce organic acids and enzymes, and they can solubilize these in non-available form of phosphorus, and thus make it available to the plant, and this fungi would absorb and make it available to the plant, so that the plant grows to its fullest, and therefore that would reflect on the yield. Also, if you see these hyphae, you can see this mycorrhizal hyphae versus the roots. The roots are much bigger in size. As far as the dimension of the roots is concerned, it is bigger than the hyphae. So therefore, it's easier for the roots to, for the hyphae to enter into the deepest crevices where phosphorus is available, and then absorb it. Whereas roots may find it difficult to enter into these crevices, and therefore it is easier for a mycorrhizal plant to provide more nutrients through these hyphae, mycorrhizal hyphae, and help in plant growth. Besides the uptake of nutrients like phosphorus or potassium, nitrogen, other micronutrients, they also help the benefit the plant in several ways. First of all, there is increased uptake of nutrients. Now it means the plant fitness. That means the plant is fit enough to, it produces more vegetative growth and then more yield. So mycorrhizal colonization would help in the uptake of, in the increased yield. In the uptake of water, uh, in places where, like on the dunes, the coastal dunes, where there is extreme water scarcity of the plants that are growing on the dunes. So these hyphae, 10% of the mycorrhizal plants on the dunes are mycorrhizal, and these hyphae, they go far and beyond, dip and then absorb the water. It maximizes the nutrient uptake, and it maintains the soil healthy and fertility. We talk about green revolution. We talk where we have used extensive amount of inorganic fertilizers, water and pesticide, insecticides, and we have destroyed the soil. So at that point of time, we were not aware that this would happen. This would be the ill effects. But then we slowly understand what have been the problems associated with the green revolution. And therefore, it is important that we have to use organic biofertilizers. And what it does is that it helps the microorganisms in the soil to thrive, multiply. So as these organisms multiply, the soil becomes more healthy and the soil fertility increases. Whereas inorganic fertilizers, you find that it will kill all these useful microorganisms and make the soil inert. Forget the fertility, it will make it totally inert and unproductive. So over a period of time, you find that adding inorganic fertilizers would mean that it will reduce the yield and reduce the soil fertility. It improves the soil structure. As I said, there is production of increase in fertility. There is a lot of organic matter that is added to the soil. Even if these mycelia, when they die, when they decay, you find that it adds to the organic matter, thereby improving the soil structure. Then it reduces the fertilizer inputs. So we don't have to add expensive inorganic fertilizers when when the plant is colonized with mycorrhiza which are efficient. Now here I would like to inform you that in mycorrhizal fungi there is no host specificity. Any fungal species can colonize any plant root, but there is something called that is host preference. A particular fungus plant would would require a particular mycorrhizal species. So like for example, if a plant would prefer the fungus X as compared to Y, then we need to identify this particular species and then add it as a biofertilizer. Then besides this, it reduces the heavy metal toxicity. This is another subject where the fungal hyphae, they can store these heavy metals. Uh, and also not only in the hyphae but other structures, they also store the heavy metals. And because of the mycorrhizal association, the plant shows better growth. And when the plant shows better growth, that means these plants which are growing in the toxic sites can accumulate more amount of heavy metals, thereby reducing the toxicity in the soil. Disease resistance is also mycorrhizal fungi. They play a very important role in reducing the diseases. One is they secrete like the phenolic substances, and then phytoalexins, and that keeps away the diseases and pests. And at the same time, the plant fitness, the more fit the plant it is, the more resistant it is to diseases. And then in case of transplanted crops, like in case of rice, or where you have to raise a nursery, when there is mycorrhizal inoculation done at the nursery level, then it will help to overcome the transplantation shock or transplantation stress. So these are a few of the benefits, but there are a number of benefits which are attributed to mycorrhizal fungi. Now, coming to these mycorrhizal fungi, how they appear, how we identify them, how we go about. Like, first of all, they would colonize the plant roots. So when we do the staining procedure, then we see certain structures which are very unique to mycorrhizal fungi, especially the arbuscular mycorrhizal fungi. And these include the intraradical, these are the hyphae which are stained with trypan blue. These are mostly non-septate hyphae you get, but under certain conditions like stressful conditions or in vitro condition, you find the presence of hyphae, the presence of septa. So these hyphae would stain blue in the plant roots. There could be other fungi also which take the stain and stains blue. But then further, the presence of these orbicles. Orbicles are hostoria-like structures which are associated, which are mycorrhizal structures, and they help in the transfer of nutrients from the plant to the fungus and fungus to the plant. So whatever the nutrients which are absorbed by the hyphae, mycorrhizal hyphae, they are transferred to the plant through these orbicles. Now these orbicles are short-lived. They survive for about a week or so, 8 to 10 days, and during the time whether they would transfer the nutrients. So when the plant is in need of nutrients, immediately these orbicles would be formed and there will be transfer of nutrients, especially the phosphorus. And then once the plant gets sufficient amount of nutrients, then these orbicles would disintegrate. Because these orbicles, they occupy quite a chunk of the host plant cell. So therefore, it would be a sort of obstruction, but it is benefiting the plant. So once it performs its function, once the nutrients are supplied, they would disintegrate. Again, these orbicles would appear. You find them during the flowering time, during fruiting time, where a lot of energy is required for the plant, and therefore these orbicles are formed where where the nutrients are transferred to the plant. Besides these orbicles, these are small tree-like structures. Then we have the vesicles. These are the vesicles which are both terminal or intercalary, and they are globose or round shape, and they help in two functions. One is they can store the lipids. They serve as storage organs, and at the same time they they form into chlamydospores during the unfavorable condition. So during the unfavorable conditions, they would form into chlamydospores. They would remain in the soil, and with the return of the favorable conditions, they would germinate. So when the plant is completing its life cycle, say annual plant, say that is the time where these vesicles are formed, and these vesicles would remain in the soil when the plant dies, and the plant also produces propagules, so that in the next season, when the conditions are conducive, you find that the propagules would grow, and at the same time these chlamydospores which are there in the soil, the resting spores, they would also germinate, and the hyphae would colonize, and there is colonization. Then you have these structures which are called as the auxiliary cells. Auxiliary cells are formed by those species belonging to those genera which do not produce the vesicles, and therefore their function is the storage of lipids. That is what we have observed also. And whereas their function as reproductive structures is not known till date. Then you have these spores. They produce spores in the rhizosphere. So they sporulate. They produce spores either inside the roots or outside the roots. So accordingly, we have the intraradical spores which are produced inside, and extraradical spores which are produced outside the roots. I mean, in the soil. When we studied these auxiliary cells in vitro, growing the spores in vitro condition and in the culture media, and then we could get these auxiliary cells and how these. I was talking about in vitro. There is septation. You can see the formation of septa, and these are the auxiliary cells. And again, the auxiliary cell, different type of auxiliary cells that you see, and we could observe the structure here. Looks like a germination, but we could not ascertain till date whether this is really a germ tube or not. But then the other function, that when we stained with Sudan, then we could get a clear staining, the black staining, indicating that they store lipid bodies. I mean, they store lipids. Coming to therefore it's easy to identify whether the plant is mycorrhizal or not. So we have to carry out a simple staining procedure and know whether the plant has mycorrhizae or not. And we know that more than 90% of the plants are mycorrhizal, and therefore very few plants belonging to certain families, they do not show mycorrhization. Coming to the classification has started way back, last 3 decades or 4 decades, and then this classification is still flexible. It is changing, ever changing, because this is the 2010 classification, how they have classified into 4 orders and different families, and then you can see here the 2013 classification. So this is bound to change in the coming days or coming years. So and it is also predicted that there are 4,000 EM fungal species on this planet, of 3,000, of which only 300 are known till date. Therefore quite a lot of fungal mycorrhizal species are yet to be identified. Now, how we go about working with these mycorrhizal uh, species is that we have to isolate the spores. It is spore based, so we have to isolate the spores from the soil. This is the rhizosphere soil. We have to sieve the soil through the different sieves. There is a procedure of Godiman and Nicholson, which we follow, and we isolate the spores. We get a different type of spores, and these spores have to be uh, trapped. That means these spores that we get in the soil, we don't know whether they are dormant species, whether they are non-viable species, or whether they are viable species. And therefore, these spores have to be, we have to prepare the trap cultures. Trap cultures is a very simple technique where we use these spores, grow them in the pots along with the host plant. The whole idea is to multiply the spore number. So whatever the spores that are viable here, they would multiply. And then when they multiply, then again we sort out the like spores, and then finally we prepare the pure cultures or monospecific cultures. So pure cultures are cultures of a single species. You can see the spores here of one type. These are spores of another type, another species. So, and these monospecific cultures are important because it helps in identification of the species. Otherwise, you end up in wrong identification, because many times taking spores directly from the soil and identifying is a very dangerous practice. It is erroneous, and therefore we need to prepare uh, at least the trap culture, so that you have a number of spores. Secondly, the age of the spore, the young spore and the old spore, they may show variation, and we should not end up in identifying as a new species. Besides, the pure cultures are used for the in vitro studies, where these can be cultured in vitro in the presence of a living root. Now, these are a few of the pure cultures that I have listed. We have about 38 species of these cultures in our polyhouse, which is dedicated to mycorrhizal uh, fungi. We have Now I'll quickly go through some of the spores of these fungi. The spores, they vary in size, shape, they vary in color, ornamentation, the wall layers, the attachments, whether they are single spores formed, I mean singly in the soil, or whether they are sporocarps are present with a large number of spores. So this is Achilospora, different genera. Then this is again Achilospora. That's what I was talking about. The Achilospora scrobicolata, a younger spore and the older spore. You can see the difference in the coloration. So if we work with the trap cultures, then it's possible to get this type of variations. Otherwise, if you directly isolate the spores, then as I said, one may end up in identifying them as two different species. This is a glomus, which has one, two, three attachments or more attachments. This is a glomus multicole, which has more attachments, and then this is a sporocarpic form where large number of spores are formed inside a sporocarp. This again funniliformis and rhizophagus species, Gygospora species, which have these type of structures which are called as the suspensor, a bulbous suspensor here also. And this has appendages, additional appendages, that is Gygospora remisporophora. It differs from Gygospora margarita because margarita is not having appendages, but you can find these nice fused laminations in case of Gygospora margarita. This is Rhecocitra. Again, they show the presence of this germination shield. I'll come to this germination shield a little later. Then this is Sclerocystis, which are sporocarpic forms. There are many species of Sclerocystis found in the soil, different soils. This is Scutellospora. Again, it shows the germination shield. You can see the germination shield here and how this bulbous suspensor is attached. This is Echinospora soloidea, the species that we have reported from our lab. It looks like a sun with the rays, and therefore it is termed as soloidea. There are some of the scanning electron microscope microphotographs of the different species of. This is another species that we have identified recently. I mean, a new species, Cephalospora felinovi, which is a species. There was only one species that was reported earlier by from Poland, from the sand dunes in Poland, and this is the second species that we reported from Goa. So it leaves. I mean, it gives a solid support that to the earlier species, and based on the different characteristics, the wall layers and all, we could identify it as a new species. So these are some of the details. The wall layers are there are only 3 wall layers here, whereas wall 1, whereas in Baltica there's other species that have been identified from Poland has got 4 wall layers. Like that, there are different minor observation that we have made indicates that it is a new species, and it has been identified as new species. These are the ornamentation, how it appears on the surface of the spore, and this is you can see the colonization. You can see the orbicles here. So the orbicles with trunk, which is quite difficult to identify, but we could use it. I mean, do it by using a specialized staining technique. And then finally, the molecular sequencing has resulted in Cephalospora felinovi, which is very closely related to Baltica, but these are 2 distinct species. Now, I was talking about the indirect germination, where we have these the germination shield. So there are 2 types of germination in mycorrhizal spore. One is the germ tube comes out from the wall layer, from the outer wall. You find that the germ tube just comes out and it forms a hyphae, I mean a mycelium, and multiplies and it spreads. The second method is the germination shield. This germination shield has got germ tube initials. You can see here, and these germ tube initials give rise to the germ tubes. So when the species which has the germination shield, they can produce large number of germ tubes. Therefore, you can see number of the hyphae coming out through these germination shields. And there's another important strategy in mycorrhizal fungi is that they spore in a spore syndrome. So they form spores inside, they sporulate inside the old spore. Now this is a strategy for survival to keep away from predators. So these spores are formed which are viable, and they remain because they are, I mean, they are kept away from the predators. So coming to the preparation of trap cultures, as I said, we use different, use either isolated spores or directly the rhizosphere soil, or seedings you can use, entire plant you can plant, and or the roots you can cut and use them in for trapping. So for trapping you have a pot with sterilized soil, and then you use the different, either of these things, any of these, and then add a host plant, that is a, we use coleus plant here. There's another technique, the funnel technique, is that you place the spores at the mouth of the funnel and then sterilize soil, where you make a slit here and put the plants like finger millet, and then this plant would, the seeds would germinate, and the roots would get into for the water. This is, we keep water in the conical flask, and the root would come in contact with the spore that is placed at the mouth, and the plant will become mycorrhizal. And then later on we transfer these mycorrhizal plants after checking the for colonization in the pots, and then we check the sporulation. You can see spores. So this is a good method for preparing the single spore culture. You can see here, this is the different cultures that have been pure cultures which have been placed in the polyhouse, where they are maintained, and you can see that the different species which are, I mean, of each one is of a particular species. So the selection of the host plant, we select coleus or finger millet depending upon the, like coleus shows very fast growth, it can adapt to the conditions, it is easily colonized, and the roots are very transparent. You can check the colonization. Large number of roots are produced. It is resistant to disease and pests, and so therefore it is a very important or a common host that we use. Besides, we also use a Eleusine corecana, that is a finger millet, in the summer months as a host plant. The different techniques that I use for inoculum production, right from the monogenic culture, aeroponics, hydroponics, pot cultures. So the most common one is the pot culture, of course, where, so coming to the pot culture, it's simple. You can multiply the inoculum from the pure culture. One can raise a number of pot cultures and multiply the inoculum, it can be used. So in case of, we have tried in different plant species like in Carica papaya, where it has helped the mineral nutrition, in increased mineral nutrition in papaya, therefore it has increased the growth. So the different species were tried. The phosphatase activity also, we find there was a higher, the acid root phosphatase activity was higher. And then this is in case of ornamentals like chrysanthemum, where we could get greater colonization when we have inoculated with the pure cultures of different species compared to the control. There was no colonization, and then number of days required for flowering also were less in mycorrhizal plants. Number of flowers increased. There was a significant increase in the number of flowers. Also in Cassandra, the similar results we could get, where we can see the control and the growth in the different, inoculated plants, mycorrhizal plants, where you can see distinct difference in the plant growth, indicating that mycorrhization helps in plant growth. This is in Andrographis paniculata, where we could see the increased growth, but at the same time uh, we could also get increased concentration of andrographolite, which is a secondary metabolite, which is medicinally important, that is used by, that is produced by this plant. In chilies also, there was the different species, the mycorrhizal species, they showed the different growth, and we could identify the Claroidium ethnicatum as one of the good species for chili. In case of ladyfingers, Echilospora scorbiculata was identified as the efficient inoculum for ladyfingers. Besides, we worked in Cereoptegal, which is a mangrove plant, And you can see the difference in growth compared to control in the different mycorrhizal plants. And mangroves are very slow growing plants. And here we could also increase the, oh yeah, you can see the colonization. The colonization, this was done in the unsterilized soil. The colonization has drastically increased in the mycorrhizal plants. And not only that, but also we could get increased root growth in the mycorrhizal plants. Even the root growth is very, very slow in the control, in the when they are 10 years compared to the mycorrhizal plants, indicating that it helps in plant growth as well as the the shoot growth, and therefore better growth in the mangrove plants. Now coming to these are the termed as the first generation inoculants, where you use the solid medium. Where you use the solid medium where the soil is used. There are quite a few limitations where you have to really maintain the cultures. Uh, you have to overcome the contamination, check them periodically, see that it works. Secondly, there could be problems like the viable, the propagules have to be, of course, even if they are viable, they can be dormant also, the spores. So when you add it to the plant, you have to ensure that there's no dormancy. And then we have also used them on the mines. So extensive work has been done on the mines where you use the mycorrhizal fungi for the nurseries. You can see also the tissue culture plantlets have been developed, and to that we could add the mycorrhizal inoculum successfully, and we could get colonization. And these plants, when we planted in the mines, they showed better resistance to the hostile conditions. So these are the nurseries where mycorrhizal inoculum was added, and these such nurseries were raised, and later on the plants were planted at the site. Well, you can see that there is a little net saving. It may not be much, but then it helps better growth of the plant, survival. It is more friendly technology than using other means of nutrients. Of late, we have been working with the Cocos nucifera. There is a coconut plant where we could develop a biofertilizer, including mycorrhizal fungi, and we could get beautiful vesicles and a lot of colonization in these inoculated plants, not only in the seedlings or these plantlets, but also in the mature plants. Where initially, when we checked the colonization, it was very, very poor. So that would help the plant. This is in the coconut roots where we could get extensive colonization, and these are some of the spores that were formed in coconut. So that is a consultancy that I have at present, working on inoculum production for coconut. This is the polyhouse where we are maintaining pure cultures, and you can see the extensive colonization in the roots of these coleus plants, and beautiful spores, very young and fresh spores, which can be used for in vitro studies. Now coming to the limitations of soil-based studies, which we have already seen, that there could be spore dormancy, the viability, and other things. Now we move on to the another area that we talk about monogenic cultures, where in vitro cultivation of mycorrhizal fungi. So by using a host, by using a plant root. So here it has large number of advantages, like there are no contaminants. You can produce large amount, quality inoculum. You can produce it in a small growth chamber with no light source. You can do the optimization of the spores by altering the conditions, determine the harvesting time. Any time of the year you can carry out the inoculum production, and once you are successful with the standardization, one can go for multiplication. So this is the root organ culture, where we use the roots, transform roots, and then the spores are used along with the transform roots, and you can get colonization, and finally you can get large number of spores in the petri plate, and you can see the spores how they look. So these are the RIPLA DNA transform roots, which have the ability to grow in the petri plates without the shoot system, and they can completely cover the petri plate within 30 days. So these spores that are produced in our culture, single spore cultures, you can see this Scutellospora scutata, and we use these spores in vitro. For the in vitro studies, you can see the germination shield here and the germ tubes coming out, and we germinate the spores. Like here also, the spore is germinated. This is Ricocitra, and then we place the spores amid the roots, the transform roots in the petri plate, and then they would grow and it would colonize the roots. This is in case of Gygospora decipiens, where we could germinate and then they would colonize the roots in the here. And after colonization, there would be there are these auxiliary cells produced. There's a lot of hyphal branching, and then you can see these are the auxiliary cells which I had shown earlier also, Gygospora decipient. And then you get the sporulation. Even in case of Gygospora, these are big spores. There's sporulation in the petriplate. Here also you can get, I mean, you can see large number of spores. There are about 25,000 spores in the single petriplate for this Rhizoglomus sclerum. And the spore, they are quite young and fresh and and glistening spores can be produced. Here also, this is another species where the spores are produced, and you can see the very clear spores produced. So here another species, there is sporulation. We could check the in vitro colonization also. Again, another species, Equilospora scobiculata, where we could get sporulation. The vesicles were used as propagules, and we could, in Rhizoglomus or Rhizoglomus clarus, and we could get sporulation using the same technique. Large number of spores we could get. Now, once we develop from the spores, now it is important that we need to know whether the spores that are formed in the petriplate in vitro condition, whether they are viable or not. So we checked for the viability and found that they were viable. And after checking the viability, we went to for the carrier. So we had to develop a carrier so that it could be used as a biofertilizer. And the carrier was prepared by using different substrates, different combinations, and we could see that the choice of the carrier also matters. The combination of the carriers also matters because if one particular combination gave us just 30% colonization, where the other was 98. Therefore, we selected this one for the studies, and then we could multiply the inoculum, in vitro produce inoculum by using coleus and finger millets, and they could be grown. And you can see the colonization. There is extensive colonization, which is mycorrhizal, indicating that the inoculum that we prepared in the in vitro condition, it is working and it is viable. We tried the same in vitro inoculum in Anacardium occidentale. There is a keju plant seedlings. We could grow them, and you can see that there's drastic difference in the growth in the when they were treated with mycorrhizal over the control, indicating that different fungi, mycorrhizal fungi, they have different effects on plants. So our idea was to identify the efficient species of mycorrhiza for keju, and you can see that colonization in keju. It showed this growth, but to ensure and confirm that there is good amount of colonization, whether is mycorrhizal, we checked the roots and found that we could get beautiful colonization, indicating that it was that means the mycorrhiza are working, and the increase in growth is because of the mycorrhizal inoculation. Then we tried with some field studies with Vigna, that is cowpea, different type of cowpea. I mean, varieties were tried, and then we used mycorrhizal species. We used a phosphate solubilizer and the rhizobium, bradyrhizobium species were used, and that's how the field experiment was conducted. And these are the seeds were treated with the rhizobium with inoculum that was prepared, and you can see extensive colonization in the inoculated plants of cowpea, indicating that the plants are mycorrhizal and the mycorrhiza is working. Now all this is done by identifying the dominant species. First of all, we have to identify which are the dominant species. Then you can always go for a consortium or a single species. We can see the effect of bioinoculants on the yield of cowpea. There was significant increase when we compared it with the chemically treated, where we used the fertilizers. The growth was substantially good, and then people prefer organic produce and has more value, get more price compared to the one that is produced by using inorganic fertilizers. This is finally the slide where a few of my students who have worked with me over the years, along with our collaborator who worked with me, and these students have really contributed. It's not exactly my contribution, but my guidance, of course, but all these students have contributed to the success of what we are doing right now in mycorrhizal research. So with this, let me thank you once again, and then now I think we are open for questions.
Yes, sir, we have few questions.
Yeah.
So, can mycorrhizae be used for hydroponic system? That's the first question.
Uh,
Can we use
yeah,
mycorrhizae in a
yeah,
hydroponic
yeah,
system?
yeah, yeah. Can be used, but we have not used. We have not used the hydroponic system. Stop sharing.
So then, what is your idea about the degradation of heavy metals using mycorrhizal species?
Yeah, when you use mycorrhizal species, like a site that is degraded with heavy metals, that site would have those metal toxicities. So there also only a few plants would grow. So those we have to identify the plants which are growing at such sites, like say where there is cadmium toxicity. So we have to identify the plants which are growing naturally at these sites. And if these plants, when we add mycorrhizal fertilizers, mycorrhizae to these plants as an inoculum, then these plants would grow better. So when the plants show better growth, would mean that the plant can absorb more amount of. So more biomass would mean more uptake of heavy metals. So these plants would store these heavy metals, and later on we can either we can use these plants for the extraction of the metals. I hope I'm clear.
Okay, so thank you. Yes, sir. So and then we have another one question. Which component in the medium is more important for isolation of endophytic fungi? Give us some selective medium to isolate endophytic fungi.
Mm, we have been using for endophytes. We have been using. Uh, I don't remember right now, but we have been using media to isolate endophytes. I've taken up this work recently, where we have identified the endophytes from the mangroves sites. I can't get it. So, but for
All right, sir.
mycorrhiza, we use different medium. Media. I can share it later on. But for mycorrhiza, we
Okay.
use the the media which are specially devised for these fungi. And when we germinate the spores, we germinate them on the media devoid of sucrose. We find that there is better germination percentage of the spores in the media that is devoid of sucrose. That media is MPN media. No, no, I'm not. MSR media for mycorrhiza. MSR media is modified Strulo and Roman media that was developed by De Klerk. And there is minimal media also. There are two media used for mycorrhizae.
All right, sir. We just have a couple more questions, sir.
Yeah,
Can
sure.
arbuscular mycorrhizal fungi diversity be affected by the climatic factors?
Yeah, can be affected because when it is when the conditions are stressful, then it can be affected, or it can be affected by the flooding. If there's more flooding, then the species diversity would decrease. Even in the toxic soils, I was talking about bioremediation of the toxic soils. They would contain there will be only few species which are able to work. It's not that mycorrhiza. I mean, all the mycorrhizal species are magicians. So few species would work. All may not survive the toxic sites, but those that are surviving and colonizing would thrive. They have to adapt to those conditions. So once that happens, then we can produce the inoculum and use it. But again, when you revert them back from these toxic sites to grow them, then they can change. They lose their ability I mean, to to tolerate the toxicity.
Okay, so one more question. What are the parameters for significant relationship between plant and mycorrhizae?
Yeah, the
The
important thing is
parameters.
significant relationship. Yeah, is the host preference. I would say that is the parameter. Host preference. A particular plant would would like association with a particular fungus. It may not do well with the other fungus. So that is what. And other things is that we normally go by the, like it could be in terms of the yield in case of cereals, or in ornamentals we go by the shelf life of the flower, the number of flowers produced, the flowering time, all these things. In fruits it could be the I mean the number of fruits that are produced, the the time taken for flowering and fruiting. But it is the preference that matters. You can use any mycorrhizal species for any plant, provided it is mycorrhizal, but it will not give you the desired results. Am I clear?
Yes, sir.
Yeah.
Thank you, sir. Your session on mycorrhizae was very informative. With all the trouble we have with the chemical fertilizers nowadays, like you rightly pointed out, it's time to look up to natural fertilizers like mycorrhizae. We wish a lot of research goes into this field in India. Thank you, sir. Thank you once again. I now
Thank you.
call upon
Thanks.
Mrs. Preeta, Assistant Professor, Department of Microbiology, to introduce our next speaker.
Good afternoon. Dr. Prabhu, Assistant Professor and Head in Charge, Department of Microbiology, Dr. ALM PG IBMS, University of Madras, Tharani campus, is the speaker for this session. He has been working as Senior Assistant Professor from 2018 till date in University of Madras, IBMS campus. He was the Assistant Professor from 2014 till 2008. He was previously working as Team Lead Clinical Research at CTS from 2010 to 2014. He was also the Teaching cum Research Fellow at University of Madras from 2009 to 2010. He did his research in molecular characterization of group C and G streptococci. He has published more than 25 articles in various journals like Journal of Clinical and Diagnostic Research, Journal of Nanomaterials, Asian Pacific Journal of Health Sciences, Trends in Biomedical, and so on. Dr. Prabhu is the recipient of Teaching cum Research Fellow by University of Madras, SRF by TBT, Research Fellow by UGC. He is currently guiding 4 PhD students, has completed different projects. Welcome, Dr. Prabhu. I hand over this session to you.
So thank you very much for a good introduction, madam. And yes, first of all, I would like to thank the management of MGR Janaki Arts and Science College for the Women, and the principal, Dr. Mani Meglai, and Dr. Nitya Kalyani, and Dr. Lakshmi Priya, and of course my junior Mahalashmi, for giving this opportunity and to present data on the opportunistic mycotic infections, which is a current topic for the discussion, I can say, because of the COVID pandemic. Let me start sharing my screen. Can you able to see my screen? Hello?
Yes, sir, we can see.
Yeah, thank you very much. So in the first discussion, like Professor Thangam Menon, my mentor and supervisor, she has given a broad outlook on this opportunistic mycotic infection. So actually, she has reduced my, job in, giving information about this opportunistic mycotic infection. But still, I'll just try to cover the important or the major points which is required for the microbiologists, I can say, because I guess everyone, the UG, microbiology people are there in this session. So I'll try to cover, whatever important in that. So why, all of this sudden, opportunistic mycotic infection everyone is talking about is because of the pandemic. Because when it comes for the fungi, I can say it is there everywhere. Generally, when people going for fungal infection for the treatment to the physician, first thing they will ask, what is your immune status? Is there any kind of activities where you had contacted any kind of activities like soil farming, agriculture, or something like that, or any injury, trauma, or something like that? First thing they will ask this question, because sometimes if there's going to be any immunosuppressive, if you are there in any immunosuppressive drugs, there are chances where you can get this upper somatic mycotic infection. And if there's going to be any trauma or injury, so there are chances where this fungi directly inoculated into the broken skin. So there's going to be a breach of immunity. So that is also going to be an important a risk factor. So and the upper somatic infection of late gained the momentum because of COVID-19. The reason for that is for COVID-19, the treatment given was glucocorticoids, that is steroids. So generally, colloquially, people will say, steroids, right? So when they're going to pump steroids like anything, there's going to be like a immunosuppression. So that's the reason where this opportunistic mycotic infection has gained the momentum with the COVID-19 patient. And yes, there was increased mortality. We used to think, Aspergillus fumigatus is a bread mold, or it may be it's seen in the fruits or some fruits and vegetables or something like that. But at one point of the time, people started reporting, saying that there's black fungus, white fungus, green fungus, right? And especially when the patients, those who are bedridden, that is in the ICU or in a ventilator, right? They started showing. And not only that, even the people, those who receive required oxygen, right? Those people also started showing this kind of opportunistic infection. And just when they investigated, they found that because of the steroids, and apart from steroids, there are going to be other factors like invasive procedures like ventilation, right? When ventilator, people, those who are not able to breathe properly, they'll just keep the ventilator, etc. Right. So that's the reason, this upper somatic mycotic infection, it just got a momentum. And yes, there was increased mortality. I should not deny that, because the reason I told you, because of the risk factor. But apart from that, I can say the environment, and somewhere else, I read in a research paper saying even a thick population can also be the reason for this, mycotic infection, not for the upper somatic mycotic infection. So upper somatic mycotic infection, I will say that one reason is immunosuppressive condition. And of late, because of our food and other habits, even this opportunistic mycotic infections are seen with the immunocompetent, that is a normal population. So for some reasons, so people are investigating the reasons for that, but still, the outcomes are inconclusive. So this opportunistic mycotic infections, right? Why this is termed as opportunistic mycotic infection is because this infection is caused by the fungi, which is considered to be the normal flora, right? Which is a normal flora, which is we used to say that in some way it is beneficial to the host, but somehow when there's going to be any breach in the immunity, right? This normal flora, that is this fungi, is becoming a pathogenic fungi, right? And that's the reason we say that this is the opportunistic fungi, right? Opportunity, where there's going to be an opportunity to cause infection. So we used to term this as an opportunistic infection. So if it is a fungi, opportunistic mycotic infection, right? And of course, what are all the conditions, different immunocompromised conditions? So one condition I told you is having a immunosuppressive drug for even COVID is a classical example. And apart from that, even for the cancer, right, and even for the organ transplant, mainly now India is a place where the medical tourism is booming like anything, because we got a very excellent medical setup for even for the surgery, even for the organ transplant. And of course, we should be very happy that Tamil Nadu is leading in our country, in organ transplantation, surgeries and other procedures, etc. So in order to prevent infection or even prevent rejection for the organ transplantation. So physicians will give you immunosuppressive drugs. So that is also a kind of a predisposing factor for opportunistic mycotic infection, because I told you earlier that this fungi will be thriving there in the environment. So this will be waiting for the opportunity for any breach in the immunity. So if there's going to be any breach in the immunity, this will come and cause serious infection. And also this opportunistic mycotic infection, if you're going to say, right, it is not only by the normal that we used to, when it comes to the fungi, we generally talk about endogenous and exogenous fungi, right? So it is not only by the endogenous fungi, even the exogenous fungi is also causing a serious opportunistic infection. For example, inhalation of spores, for example, the opportunistic mycotic infection like aspergillosis, cryptococcosis, and histoplasmosis, right? Can be transmitted with inhalation of spores. And of course, candidiasis, this is a endogenous fungi, I can say, which is there in all your mucocutaneous region, right? Then and there, it causes serious infection. For example, in HIV patient, if you're going to see, you can see this oral thrush where, which is there in the tongue. And not only that, right, sometimes we used to get food poisoning, right? Sometimes you used to get food poisoning whenever, when you're weak. Sometimes people will say that when you're going to a physician, so just boost the immunity. That's the reason, you are frequently getting, fever or diarrhea or any other, symptoms, right? So what is the reason, when your immunity is getting down, what is the reason you're getting, frequent infection? Is there's going to be a breach. This is a kind of, a breach in immunity, I can say. Right. So when you're going to take contaminated food with the fungi, right, or any mold or something, like when you're going to take that, then when there's going to be any compromise in your immunity, then you're getting a serious infection as food intoxication. That is happens. So you must be wondering, see, when somebody's going to a restaurant or a hotel there, where, there'll be 10 number of people will be having, but among the 10, one will be having a severe, gastrointestinal symptoms. But all the 9 will be hale and healthy. So we cannot blame the restaurant, right? Right. So I'm not saying that, restaurants are good, right? Sometimes, when out of the 10, that one people, right, one person, if they have a poor immunity, there are chances, they can develop, symptomatic, symptomatic conditions, and that's the reason out of 10, one is having an infection, kind of, especially diarrhea or something like that. So immunity, which plays a vital role, when it comes for opportunistic infection. And apart from the, the transmission, or inhalation, or the source of infection, the kind of infection also plays a vital role, right? The infection ranges from mild superficial to systemic infection. Don't think that this opportunistic infection for mycosis, it just causes only a skin infection or something like that, because many people think that okay, fungal infection means only it causes skin infection, right? All your advertisements, TV ads, and even in the social media ads saying that skin infection means they are saying only about the fungi. No, there are life threatening conditions caused by opportunistic fungi, which is very commonly seen in the immunosuppressive patients. So it is all about the it starts with the superficial, and even it just enters into the blood, where it can cause a serious systemic illness. And yes, the example I can say is candida, it is endogenous fungi, where yeast, more particular, it can cause mucocutaneous to cutaneous and systemic illness. And there are fungi, like aspergillus, which can cause allergic like reactions. Many of the time, we used to get allergy like reactions, skin rashes. But what we used to do is we just go to a medical shop, over the counter, we just get an antihistamine, and we used to pop that antihistamine, and then 3 days, by the time, it will just go off because of the immunity. It is not because of the, antihistamine. Antihistamine is just to reduce the allergic reaction, but it is not an antifungal agent. So what I'm saying is, aspergillus can also cause allergic reaction, ranging from allergic reaction to allergic bronchopulmonary aspergillus, which is a very serious condition where it just affects your lung, I can say. So when it comes for opportunistic infection, you need to consider whether the infection or whatever the fungi you are isolated from the infected side, like whether it is a colonizer or seriously, like it is like causing an infection or a disease. So that depends on the, depends on the microbiologist. So the role of microbiologist is going to be very, very important in that condition. Because just like that, people will say that, you're just doing something which is, like isolating the bacteria, fungi, or something like that. No. So the role of microbiology is very crucial in managing and treating the patient, not only with this opportunistic mycotic infection, even in the emergency conditions, right? Like if they are asking for any blood culture or any serological thing, like based on that only they'll go for the treatment. So you should be proud that, like you are doing such a wonderful course, like in a life-saving course, I can say, which is equivalent to a medicine, because based on your reports only, the doctor is going to, like treat the patients. So that is a very, very important thing. So generally for a clinician, it will be challenging to decide on that. So they'll be sending the samples to you, and it is up to the microbiologist to take a call on that based on the clinical case history, right? Like you need to decide whether you need to go for a 1 sample, 2 sample, because morning Dr. Tangamenan ma'am was talking about, like how to do for the sampling. It's not like in 1 sample you can fix the the fungi with responsible fungi. At least you need to go for 3 samples and check for viability, and then you can confirm that okay, fine, this is due to this, because I told you previously that this fungi thrives in the environment, so there are chances where like contamination may happen if you are not collecting the samples. So at last, like this antifungal agents also, which is very, very important. But unfortunately, what happens, right? Because of the antifungal resistance now, even treating with the antifungal agents is also very challenging. One thing is, like toxicity. The second thing is, like the resistance, which now even the microbiologist is facing, because not many labs in our country where like they are doing routine antifungal, susceptibility testing. That is very important when it comes for treating and managing this opportunistic infection. So I was talking about predisposing factors. There are many predisposing factors. So so there are 2 important factors you need to consider, right? Under this, immunologic factors and non-immunologic factors. So immunologic factors, I was talking about this immunosuppressive, right? So it is not only by the immunosuppressive drugs, even by the conditions, I can say, where for example, HIV, which just makes the patient immunosuppressive, and of course cancer, where because of the chemotherapy, sometimes the patient will be immunosuppressive, and of course neutropenic patient, right? Solid. See, all those patients are also very important. So those factors are considered to be the immunological factors. And what is the non-immunological factors are like steroid abuse, right? And one thing I just want to insist is this diabetes. So now India is kind of a diabetic capital, right, where everyone is popping this metformin, that is anti-diabetic drug, and because of the food and lifestyle, right, almost 40% of the population has become a diabetic developed diabetic condition. That also favors the opportunistic infection. So you need to consider all these factors when you decide or when you think about this opportunistic mycosis. So why there is a trend, because everyone is talking about opportunistic mycosis. After COVID, there's a lot of awareness about this opportunistic mycotic infection among the physician, because previously they thought it is a mold, right, a laboratory contaminant. Otherwise they'll say it's a contaminant. Maybe you would have not used you would have not sterilized the plate or test tube, or maybe the place where you are working, it's not a sterile area or something like that. Right now, after COVID, they realize that okay, fine, this mycology people are very serious guys, crazy people, right? We need to depend on this people. Otherwise the patient, it is very difficult to treat and manage the patients, right? So the clinical awareness which has happened after COVID-19. So there is a lot of talks is happening about this opportunistic mycotic infection, and of course improved clinical diagnostic tools. So one good thing about this COVID-19 is even a common layman now, they are talking about real-time PCR. So I'm happy about that. right? Like, because previously before COVID-19, hardly 1 or 2% of the population, which who are working on the field only, they know what is real-time PCR, what is use of real-time PCR in diagnostic setup and etc. But now after COVID-19, no, everyone, I can say 80% of the population now, they are aware the role of this real-time PCR in diagnostic in infection. And thanks to that real-time PCR and other molecular techniques, where now it's very easy to go for diagnosing this opportunistic mycotic infection, because very well that when it comes for mycology, it takes time for time of inoculation to the growth, right? So usually people will say that mycologists are the laziest people, because it takes time, right? Because you inoculate 2-3 days after, you just go for the incubator and see whether the growth is there or not. Otherwise, all these 3 days, they need to sit idle, and they need to do other activities. But yes, of course, we have state of art facilities and techniques which are available now, where in an hour, you can just detect the fungi which is responsible for the infection, etc. And of course, the increase in the susceptible host is something where a rise in diabetic conditions. That is, I told you that almost India is going to be the diabetic capital in a year, where majority of the population, that is 30 to 40% of the population, will have this diabetic condition. So that is a risk factor for this opportunistic infection. So that is also one thing. And second thing is invasive diagnosis. Now I told you that India's India's medical tourism is booming because of good doctors and hospital in healthcare facilities and everything. So more and more healthcare facility, more and more advancement in the medicine, what happens is more and more invasive procedure will come. So that invasive procedure is also a risk factor, because I told you that this fungi will be thriving in environment. So there are chances where this can get into the tissue or any other place where this surgery is happening. So necessity to know, this is what I was talking about, because after COVID-19, everyone is talking about opportunistic infection, because COVID-19 infection just makes this harmless fungi to become a pathogenic fungi if there's going to be any breach in the immunity, right? And of course, there are many co-infections are happening even now in after COVID-19. There are many case reports saying that they're having this kind of fungal infection, that kind of bacterial infection. But still, the scientific community is doing a research on the COVID-19, post COVID-19 infection. What is the reason for getting this kind of bacterial and fungal infection? And of course, three important fungi, right? Candida, Aspergillus, and Mucorales, family, that is Rhizopus, Mucor, and Rhizomucor. These three important opportunistic fungi are considered to be the very important one because of the COVID-19, I can say. Because previously, no one bothered about that. Because after COVID-19, they were saying that black fungus, yellow, green fungus. Now, everyone is talking about that because of the COVID-19, right? And this Candida, Aspergillus, and especially this Mucor, right? Mucorale family group of fungi, and next to that is this Aspergillus. These two fungi are the very deadliest, I can say, during the time of COVID-19, because even after escaping from the COVID-19 infection, people started developing infection, this kind of opportunistic fungal infection, and many people died because of this, because of lack of awareness, I can say. But off late, right, now I told you that there's a lot of awareness happening with those clinicians, so we are happy about that we can able to treat and manage this kind of infection with the patient. So when it comes for the pathogenesis, because I told you that this is a harmless fungi, how it is becoming a pathogen, right? So when there's going to be a breach in immunity, it's not like that only during the time of immunosuppressive condition, it just produces the enzyme toxin or other things. No, it just produces during the a previous stage also. Only thing is our immunity is very good, so that it just removes those toxin, enzyme, and other pathogenic factors. But when there's going to be a breach in immunity, then the problem starts, because all these enzymes and toxins are very deadliest, like I can say for example, we're going to see enzymes like phospholipase, proteinases, and hydrolytic enzymes. These enzymes which are very very important for, these enzymes are produced by candida, for example, right? Like it just helps this yeast to host tissue invasion. That is a very important factor. And apart from that, dimorphism. So dimorphism is for example, if you're going to take yeast, like it has got two phases. One is yeast phase and mycelial phase. So yeast and mycelial. So mycelial phase, if you're going to see, like it is very important phase in the tissue invasion. So if you're going to take sample from the tissue, right, like you can able to see only the mycelial phase, because this mycelial phase is responsible for like producing certain hydrolytic enzyme that is acting as a spreading factor. So that's the reason where this mycelial phase is very common seen in the tissue sample. And apart from that, different cell morphology, like a smaller one, bigger one, because if you're going to see as if like aspergillus, you got fumigators, flavors, Niger, etc. But why only the fumigators? They are targeting fumigators because fumigators the one which causes serious infection, right? It is very commonly isolated from the COVID-19 patient. The reason is because of the smaller spores. It's not like that, flavors doesn't cause, like a serious opportunistic infection. Yes, it causes serious opportunistic infection, but the anatomical region is different. For example, flavors, if you're going to see, like it just restricts its infection with the sinus, sino-orbital region, whereas fumigators can reach even to the lungs where it causes serious infection. That is also there. The reason is the spores of fumigators are very small, whereas for the flavors, it is very bigger one. And apart from that, like we have other factors like environmental and pH, of course, like pH is also there, and mutation, right? Mutation for drug resistant, even not only for the drug resistant, even for thriving in the harsh environment, that is also there. And cidrophore biosynthesis, cidrophore production, and amino acid biosynthesis, right? Like all these things, which just contributes to its pathogenesis, in the sense, like not only to affect the host, in order to also for evading the host immune system. Immune properties or something like that, even complements, opsonization, because those complement proteins are very very important in eliminating this kind of fungal infection, where there's going to be any like breach in immunity, or if there's going to be any other factors like this factors, I can say, then yes, this is going to play a major role in causing serious infection. And apart from that, biofilm formation, and especially this biofilm formation is very common in the invasive infection, because there's going to be any invasive procedure like catheter or any other procedure, right? So what happens, your catheter will be there in your organ, and that actually it reduces the immunity. So that the chances where if your environment or even the catheter is not sterilized properly, right, like if it is unsterilized, or even it may also carry, when there's a time between opening the catheter packet and putting that into a body, right? So in that time, the chances where this environmental fungi can sit on that catheter and can cause a serious infection. And how it is escaping from the antifungal agents or antimicrobial agents is because of the biofilm, where biofilm is nothing but a kind of a micro colony of organisms, where it's just communicating with the organisms, so that it just cover, I can say, where it just protects from the immune cells, and also from the antifungal agents. And apart from that, we can also talk about capsule, right? Because capsule is the outer layer. This is also seen in the bacteria, right? So this is the outer covering which evades a phagocytosis process. And then immunology, this is what I told you, right? Because immunity plays a vital role, right? So people, those who are immunosuppressive, are considered to be the risk population. So when there's going to be any breach of immunity, then it's going to be a risk for getting this opportunistic mycotic infection. So both the cell and innate and cell mediated immunity is going to play a vital role. For example, if you're going to take a normal person, this innate and cell mediated immunity, all your neutrophils, all your complement proteins, all your lymphocytes are going to help you. T cells, B cells, everything is going to help you. But when there's going to be any immunosuppression, this fungi will be happily thriving there in your in your tissues, and yes, it causes serious infection. And that's the reason I told you that this immunity is going to be very, very, very important for the opportunistic infection, and of course genetics. So must be wondering, in a group of 10 family, right, like even in the with the COVID also, like a 10 member in a family, right, one will get a COVID infection. So max to max 2-3 people will have the infection, but remaining 7, will be, like very happy, I mean, hale and healthy, and they will not have any infection. They will not be symptomatic, at least, right? Like they'll be asymptomatic. And the reason is, like genetic. That's what, like people have discovered that, like it's not like all the 10 people in a room will have an infection, right? Like there are chances where, like 4 to 5 people will be symptomatic, and remaining 4 to 5 people will be asymptomatic, right? So the reason for that is, like genetics. In a very similar way, even this opportunistic fungal infection, it is happening with not it is not happening with all the like immunosuppressive patient only, but the few immunosuppressive patient or a diabetic patient, I can say is the reason could be genetics. And this genetics can be anything. So the genetics in the sense, there could be any mutations, or there could be any other factors, but just gives a protection against this opportunistic infection. And generally, like we used to term that as any genetic variation, maybe a single nucleotide polymorphism mechanism, like any, it's just going to be any change in the like single nucleotides, right? Like any base change, like that actually makes makes the people like either immune or not immune to the opportunistic infection. So the real troublemakers are Candida, Cryptococcus, Aspergillus, Rhizopus, and Mucor. So Cryptococcus, so far, like they have not reported with the COVID-19. So since because I included this Cryptococcus, because other immunosuppressive conditions, this Cryptococcus plays a vital role. So candidiasis. So this is a most common, like opportunistic fungal infection in developed world, I can say, especially with the immunosuppressive patient. And if you're going to see, like this candidiasis is the fifth, largest, fifth largest infection, I can say, fifth infection or fifth largest infection in ICU, especially this pediatric ICUs and all, right? Like because the chances where, like they're not going to decontaminate. See, if you would have seen when you're collecting the blood, or when they are putting a needle inside, I mean, for taking your this dextrose or NS and all, like they're going to in case, like if you're going for the hospital, right, like when you're getting dehydrated, they'll go, colloquially they'll say that, glucose, take the glucose or something, so when you're going for, like intravenous, right, like what happens, right, if they are not decontaminating, right, like, or sterilizing that area with this alcohol, that is 70% ethanol or spirit, or any other, like disinfectant, there are chances where, like this candida which is there in the skin can be go, it just go inside the bloodstream and cause a serious infection. And that's the reason they are saying this is a fifth infection, largest infection, I can say, with this ICU, and especially with the pediatric patient. And just, this is all kind of an hospital acquired infection, because I don't think this this opportunistic infection only because, like happening with the immunosuppressive patient. This can also be a nosocomial infection, because many of the patients in the hospitals are very weak, and they'll be very they will have a very poor immune system, and they'll be with the different drugs. And of course, there are patients who have undergone surgery or invasive procedure, they are coming under this population, and those population can have this kind of this candidiasis. So just, we should make sure that, like we are sterilizing the area of surgery or intravenous like procedure, I mean inserting your needle, or right. So just make sure that, like you're putting your sterilizer properly, otherwise there are chances of getting this candidiasis is very, very high. So candidiasis, you're going to see, right, like so what we used to study in our academic side is, like only oral thrush, or like vaginal thrush, etc. But apart from that, like this candidiasis, this candida can also cause invasive candidiasis. So I told you that, like through intravenous inoculation or through surgery, this can enter into the bloodstream and cause a serious infection. And also it can also cause deep-seated infection when it comes for like surgery, right? And That could be, even without candidemia. So there's a surgery part, right? So that is also there. So when it comes for candidiasis, there are three forms of candidiasis. So morning madam was talking about that. There is oropharyngeal, esophageal, and vulvovaginal disease that comes under the mucocutaneous candidiasis. So this 80% of the mucocutaneous candidiasis is responsible. Who's responsible for this? Candida albicans, right? And remaining is non-candida albicans. So we have Glabrata, cruzi, and we have tropicalis, right? So now there's much talks is happening about Candida auris. So morning madam was talking about that Candida auris. Uh, you must be wondering, so where all these organisms are coming from? So all these organisms are there. Only thing is, when there's going to be any breach in immunity, or is there going to be any kind of activities like surgery or immunosuppression drugs or something, then yes, we started getting this kind of infection. And not only with this kind of invasive candidiasis, even other candidiasis, like oral thrush and vaginal thrush, even that is also reported with the HIV patient. So I'm not touching that part, right? Because even in the HIV patient, even the cancer patient, this oral thrush is very commonly seen, right? And a neutropenic patient, of course, those who are receiving a transplant, those patients are also are coming under the risk of getting this infection. And Candida glabrata appears to be the most common Candida species in malignancy and transplant, whereas Candida cruzi is among the hematologic malignancies who receive fluconazole, prophylaxis. And there are chances where the fluconazole resistance Candida cruzi is, getting this Candida cruzi is very high with this because of malignancies. Next is Cryptococcosis. My favorite. I did my PG dissertation in Cryptococcus, not Cryptococcosis, isolating the Cryptococcus neoformans from the trees. And this is a very common fungi, everywhere. This is also environmental fungi, and the source is like pigeon droppings, I can say, and pigeon droppings, and also wherever the niches are there, right? Tree niches are there. So there, you can able to isolate this fungi. And unfortunately, this Cryptococcus neoformans is also isolated from the immunocompetent host. And the reason for telling this is usually the immunocompetent host, they will not show any symptoms for Cryptococcus infection, and that's the reason the mortality is very high in the immunocompetent patient. Right. So for the immunocompromised, at least they'll be symptomatic, and the symptoms will be very similar, very common. Like you will have some altered mental status, lethargy, or memory loss, or something like that. But still, that will be the sign of like cryptococcal meningitis. Usually, they'll go for CSF for getting the information about the cryptococcus, like whether the cryptococcus is there or not. But when it comes for the immunosuppressive patient, this Cryptococcus neoformans is very commonly isolated, and whereas the immunocompetent patients, right, like Cryptococcus gatti. So far, whatever the reports, which I got it from the literature. So this was the report. So don't think that only with the HIV patient, because when it comes for Cryptococcosis, when I did my PG that time, people are telling me that I think when I'm going for a sample collection and etc., they are talking about this Cryptococcosis, saying that this Cryptococcosis is only seen in the HIV patient. No, absolutely no. This is also seen with other immunosuppressive patients, such as solid organ transplant recipients, etc. Right? And of course that's a different story. It is not like that. This all the solid organ transplant patient will get this infection. Only 7% of them will get this infection. That is the different story, I can say. And sometimes this infection can be like after 1 or 2 years after the transplantation, because for some reason, right, what I found in the literature is, one thing is either this will be in a latent state, right, but even I'm not sure, still it is inconclusive, because in one paper they are saying that other factors, other like melanin and other factors are there, And some people, they're saying that, like maybe just, maintains a latency. That's the reason, it just waits for, after 1 or 2 year, like, yes, it just, the patient started showing the symptoms of cryptococcosis. Right, that is also there, and there are reports saying that this cryptococcosis may enter into a blood, can cause a disseminated infection, and can affect other organs, and of course, like, this is also commonly, like, reported in the diabetes, or very rarely. Don't think that disseminated cryptococcosis is seen in all the diabetes mellitus patient. So whatever I've like mentioned here in this presentation is purely, like, it's based on the research papers, not from any textbooks or something like that. Then it comes to the aspergillosis. So I told you aspergillosis is just aspergillus, which is there in a environment. When you're going to inhale the spores, it just goes to the lung where it causes serious infection, right? So because of the inhalation of the spores, like, we are getting this infection. And aspergillus fumigatus, because of the smaller spore, like, it just goes to the lung, whereas the flavus, which is a very bigger one, right? The spores are Cordycea's bigger one. So that's why it just stays in the sinus tract, where, like, it just causes sino-orbital infection. And this, even the aspergillosis, like, two types of aspergillosis. That is, one is non-invasive and invasive. So non-invasive, it can cause allergic bronchopulmonary aspergillosis and allergic fungal rhinosinusitis, whereas invasive forms, where, like, it can cause a chronic pulmonary aspergillosis, and of course invasive pulmonary aspergillosis, where it causes aspergilloma. So it'll be almost looking like wherever it ends with oma, right? We used to say that's a cancer condition, right? So it'll be looking very similar to that, but no, not it is not a cancer condition. It's because of the aspergillus, because of all your immune cells which is getting deposited there. That's why it is looking, the tissue, in the tissue, it is looking like that. So that's why we are saying it is aspergilloma, right? And this aspergillosis, if you're going to say, this is very commonly seen with the patient, those who have admitted in the intensive care unit, and people, those who have a severe respiratory viral infection, for example, COVID-19, and of course, the patient, like, with immunosuppressive therapies. Then it's a mucormycosis. In this, you have a rhizopus, mucor, rhizomucor, right? Like this, all this, which contributes 75% of the mucormycosis cases. And next to aspergillus, this mucor, this rhizopus, mucor, and rhizopus, contributes the majority of the invasive fungal disease in the patients with malignancies or the transplantation, I can say. And when it comes for mucormycosis, the diabetics, even with the COVID-19, if you're going to see, right, the diabetic people are the risk population of getting this, black fungus. They are talking about black fungus, right? So mucormycosis for some reason, right? And next to that, diabetic people, like the people, those who have a trauma or burn injury, are found to be the risk population, right? And for some reason, like this rhizopus and mucus, there's a big, like factors and other, things which are involved in, like causing the serious infection with the diabetes, which I'm not going to touch on that, because that itself takes a separate topic, I can say. Just you can have an idea about this. That's it, right? And generally, mucormycosis, mucor, rhizopus, and which is there everywhere in the environment. When you're going to inhale it, yes, you're going to get this infection. So the normal person, right, like they have a very good immunity. That's why it is getting removed. When people, those who are having immunosuppression or immunocompromised, then I think, like this causes serious infection. So madam is talking about in the morning, rhinocerebral zygomycosis, pulmonary zygomycosis, cutaneous zygomycosis, gastrointestinal tract zygomycosis, and disseminated. So everywhere, because this fungi is there in the environment. So then and there, if there's going to be anything that is happening in your body, any trauma, injury, or any invasive procedure, surgery, or immunosuppressive condition, yes, this can cause a serious infection, right? And if you're going to see the healthcare associated mucormycosis, right, like that could be a reason, where, like the majority of the COVID-19 patients were getting this this black fungus infection. The reason could be like using a non-sterile products, like because they were using this oxygen cylinders, right, like so I read in papers saying that like using the oxygen cylinders, like which is not maintained in a proper condition, because that was the time there's a lot of like scarcity for the oxygen thing. So they were just taking the oxygen cylinders from the industry, and like they were just I'm not sure like this I mean, this report is true or not, but I read in a paper saying that like because of the unsterilized oxygen, like the or not kind of a commercial cylinder oxygen cylinders are using for the patient. That's the reason where like people started developing this kind of infection. I'm not sure about that, but there's one paper like stating this. About the like non-sterilized products for the and of course like there are chances where like if you have a defective ventilation system, right, like an environmental factor, like, so that's the reason where like during the COVID-19 where people were saying that they're not to use your ACs, and because ACs are the thing where like that can spread the infection or something for the virus. But still, like spores are spores, right, like it is not only for the virus, even for the spores I can say. Because uh, I just want to say one thing, that once I just went for one inspection to a college, saying that the college chairman was asking me why microbiology laboratory requires air conditioner. So I was just explaining to him that like, sir, it is not for the people those are working in the like in the lab. It is only for the safety of the people who are working in the lab, because when you're going to put a fan, there are chances where in this pores can spread easily to the other, because we are working with the bacteria, fungi, right, any other thing. Because we cannot judge the people, I mean the person immunity by the appearance, right, like, so they may be looking well, but when it comes for the internal immunity, chances they may have a very weak immune system or something like that. So for that reason only we have ACs. So ACs will give you a good temperature, but at the same time it will not allow the spores to spread, right, like that's the reason, right. So environmental factors, and yes, the ventilation system, that is also very, very important, where like when it comes for like healthcare associated mucormycosis. So that is all about the thing. When it comes for the diagnosis of infection, I told you that like why people started like talking about opportunistic infection is because we have a state-of-art facility for diagnosing the fungal infection. Because previously only we had a plate, like a SDA plate, a PDA plate, where we need to put the sample, we need to wait for 3-4 days, like to get the results, etc. But now we have a state-of-art facility where, with the help of real-time PCR or a PCR, where in a molecular techniques, any other molecular techniques, in 2-3 hours, like you can get the result. We can go for the patient, treat the patient, and 2-3 days, like you can just get out of the hospital. Otherwise, what happens, right? Like if we are going to put the patient in the hospital, right, that is also a risk factor for the opportunistic infection, because long hospital stay can also be a risk factor for the opportunistic infection, right? So with the help of this like molecular diagnostic techniques, we can just like get the result, treat the patient, send the patient happily to the home, right? But still I will say that like gold standard is like using as Sabros dextrose agar, right, where like you can able to at least like like characterizing the fungi, what fungi it is, and like and whether it is resistant to antifungal agents, etc. So apart from that, like we have radiology, because morning madam was talking about like glass ground appearance, right, like because every fungal infection has got a unique like appearance there in the X-ray. So based on that also, that is clinical, right, like so clinical along with the radiological evidence we can say. But still like conventional mycological methods are very, very important, and especially the microscopes, which plays a vital role, because without LPCB mount we cannot say that like this fungi is this or that or XYZ or something like that. Because when it comes for like especially this mucorales family, right, like everything will be looking similar. So you cannot come to one conclusion based on seeing the like colony morphology or the moles, like what is there, right? You cannot come to one conclusion. At least like you should go for a microscopy and an LPCB mount there, like based on their conidia and the conidiophores and etc. Yes, you can just like diagnose the fungi, and especially the opportunistic fungi. So therapeutic decisions. So why why I just put the slide is like how a clinician is going to take a decision whether the isolated fungi or identified fungi like it is because of this, that can cause, that is responsible for the infection or something. So always, like a patient will come with the atypical signs and symptoms when they have an opportunistic infection. So clinician should consider everything. And of course, when they go for a tissue sample, histopathological, staining and everything, there, like they will have unusual reports. And also the fungi will have unusual affinity to the different organs, right? Like you would have read in a paper, a paper, not in the textbook, saying that like this organism will be restricted to only this organ or something. But when it comes for opportunistic mycotic infection, this can be anywhere in your organ or something like that. So prevention, just avoid exposure to the fungal. So if you think that you are immunosuppressive, if your immunity is very weak, just make sure that avoid exposing to the fungal spores. At least now we have an awareness about this, putting a face mask and everything. So when you're going outside, wear a face mask. If you think that you are under a risk population, like diabetes, renal transplantation, or solid organ transplantation, or immunosuppressive drugs, or neutropenic patients, right? Just make sure that we are wearing a mask and going outside, and and appropriate treatment, that is also very, very important, right? Treating with the right antifungal agent, that is also important. So before that, going for antifungal susceptibility testing is also very important, preventing serious condition, I can say. So treatment, we have a wide range of treatment. For aspergillosis, we have amphotericin B, itraconazole. For candidiasis, we have amphotericin B, fluorocysteine, itraconazole, nistadinazole, clotrimazole, right? And of course, for the cryptococcosis, we have amphotericin B and fluconazole. So like this, we have. So for COVID-19, they were giving COVID-19 patient, they were giving lipophilic amphotericin B, right? That is the only drug. The problem with the antifungal drug is, they are more toxic, and that's the reason where it should be given under the prescription of physician. Otherwise, just like that, you cannot go to a medical shop and get this amphotericin B or any other antifungal agents like fluconazole, itraconazole. Even if you're going to see the dosage and all, they'll give a weekly once or weekly twice or something like that because of the toxicity, right? So conclusion, I can say, is this opportunistic infection can be prevented, right, by special measures like good personal hygiene and early and regular medical examination of the risk population, and of course, good diagnosis and appropriate antifungal treatment can actually, avoid opportunistic infection, and also, decrease the mortality and morbidity of this infection. So thank you very much.
Thank you, Dr. Prabhu. We have few questions. Is there any ethical clearance required to work with opportunistic fungi associated with COVID infections?
Yes, it is required, because you are collecting a biological sample, and you should have a proper containment facility, either BSL-1 or BSL-2, and you need to present before the ethical committee saying that how we are going to collect the sample and how we are going to process the sample, and finally, that is very important, how we are going to dispose the biological sample.
Okay, sir. Next question is, how can we overcome the fungal diseases, especially while working with opportunistic mycosis? Is there any special precautions that we need to take when we are working with opportunistic fungal infections?
You mean to say that research or in the lab?
Yes, sir. So that is what they mean, I think, in the lab. When we are working with the opportunistic in the lab, of course.
So very basic thing only, I will say that wear mask, glove, and just work in your containment with either BSL-1 or BSL-2, right? That is more than enough. And proper disposal of your biological material, that is also very, very important. Otherwise, second thing is, just make sure that, students who are working in opportunistic infection, just make sure that, you don't have any other like health issues, like or any other, like you are under the treatment like immunosuppressive drugs or etc. Just make sure that you don't have anything. Otherwise, like very well you can work with the opportunistic fungal agents if you have a good infrastructure.
Okay, sir. So then we have a couple more questions. What is the role of biosurfactant producing fungi in the environment? I think maybe,
I'm not
is
sure
it
because
relevant for your topic, sir?
no,
Is it
no, I'm
relevant
not
for
sure
what is
because
it?
I'm
Yeah, yeah.
so into its medical microbiology. So
Okay,
maybe this
fine. I think this is
biosurfactant.
not a relevant question for you. We'll move on to the next question, sir. Sorry for that. How can retrobulbar amphotericin B halt the disease progression of rhino-orbital mucormycosis?
Even I am not sure about this because retrobulbar amphotericin B. Maybe they are talking about like giving this amphotericin B. Like in uh, you know uh, there's something called we have animal experiments retroorbital, right? Like maybe are talking about that. I'm not sure about this question also, retrobulbar amphotericin B. But generally, like amphotericin B, when they are going to give us IV, like it has got a very good like activity. I mean, against like this apoptotic mycotic infection. I'm not sure
Okay,
about
thank you,
this
Dr.
retrobulbar. I'm sorry.
Yes, sir, that's fine, sir. Thank you, Dr. Prabhu, for explaining opportunistic infection in a very simple term and educating us on all the aspects of fungal infections. It has been a pleasure for me personally for hosting my senior session today. Thank you, sir.
Thank you, Mahalakshmi. Thank you very much.
Now we have Dr. Sai Subhashini. She would be introducing our final speaker, Dr. Arvind Prashant.
Dr. Janaki College of Arts and Science for Women, I cordially welcome our chief guest, Dr. D. Aravind Prasad, Associate Professor, Periyar University, who has 14 years of teaching and research experience. He is in the he is in charge of Periyar University Microbiology Club, Fleming Journal Club, coordinator for the Health Awareness Program, and member of the selection committee for MPhil and PhD. His area of research is endophyte biology and antimicrobial resistance. Under his guidance, 5 PhD, 23 MPhil, and 53 MSc students completed their research studies. He published his work in more than 25 international journals and presented more than 75 papers at a conference. He was awarded Dr. APJ Abdul Kalam Memorial Award by the Society of Scientific Research. His paper was awarded second place in the oral presentation at the International Conference on Food, Energy and Water Microbiology. Today, he is going to deliver a special lecture on endophytic fungi, current scenario and future prospects. We are grateful to have him on the stage. I now invite him to enlighten the students with this knowledge. Thank you.
Thank you, madam. Thank you for this nice introduction. Am I audible now?
Yes, sir, you are audible, sir. Sir, you are audible. Arvind, sir?
Am I audible,
Yes, sir.
ma'am?
Yes, sir, you are
Yeah.
audible, sir. You are audible, sir. But we are not able to
Yeah.
see your video, sir.
Okay. I'll switch on my.
Yes, sir, please.
Yeah. Yeah. Kindly help
Thank
me
you.
in sharing the screen. Okay. Let me. Um, It's a slide visible, ma'am?
Yes, sir, the slide is visible, sir.
Yeah. Okay. Thank you very
It's
much
not
for
in
giving an
the
opportunity.
presentation. Sorry, sir. It's not in the presentation mode, sir. I can see the introduction slide.
Okay, okay, 1 minute. 1 minute.
Yes, sir.
Now it is okay?
Yes, yes.
Thank you
It's
very much
fine,
for
sir.
giving. Yeah, yeah. Thank you very much for this wonderful opportunity for presenting my few of my research findings. As you are aware that in the morning, people have taken about the fungi as a villain. And this is a new topic where we are going to switch over to endophytic fungi, the current scenario and the future prospectus. And this is a picture of our Department of Microbiology, Periyar University, Salem. And my laboratory is located down where you can see my students working medical microbiology laboratory, where we are concentrating mainly on endophytic fungi. Right. Many people would have not known about the definition of endophytes, or this has been newly introduced. In 1866, D. Berry introduced the term endophytes for those microbes which reside within the plant of healthy tissue of the plant without causing any apparent diseases. And initially, Carroll in the year 1986 coined the term endophytes for those organisms that causes asymptomatic infection within the plants. And later his definition was replaced by Petrini in the year 1979 as those fungi which live in symbiotic relationship within the plant without causing any apparent infection in the plant. It includes both fungi as well as bacteria. Later, over the passage of time, the definition of endophytes was defined and were remodeled, and now it has been those fungi which resides within the plant without causing any apparent harm to the host plant and live in symbiotic relationship with the plant. These endophytes enhance the host growth, nutrition acquisition, improve the plant's ability to tolerate abiotic stresses such as drought resistance, salinity, and enhance the plant resistant to insects and pathogens and herbivores. In return, these organisms, in return, draws the nourishment from the host plant, acquires the character of the host plant, and starts producing the bioactive metabolites. When you talk about these endophytes, initially these endophytes were not considered until the advent of the Taxol, an anti-cancer drug that was first discovered from an endophytic fungi. When you talk about these endophytic fungi, the plant which is chosen forms the major aspects. Plant growing in areas of great biodiversity have the prospects of housing several endophytes. And usually the fresh part of the plant is preferred when compared to the older tissues, which often contain many fungi. And when you see the literature, uh, not only the plant, any part of the plant, like the leaves, flowers, and fruits, are effective. And not only that, the stem and root are also very effective in the isolation of these endophytic plant. When you talk about this endophytic research, how far this endophytic research can be beneficiary? Nowadays, people are facing a problem of antimicrobial resistance. And now we are in search for a new compound or a new drug which has been effective against various organisms. Organisms have started evolving rapidly, slowly, step by step, and now they have become very resistant. So there is a need for a new compound, and the shift has been made towards the medicinal plant, which are now the only source of bioactive compounds. And hence, when you talk about these medicinal plants, we are not in a position to utilize all the medicinal plants, because once we start destroying these plants, ultimately it will result in the biodiversity imbalance. So there is a need for the other set of organisms or the other opportunity to go for these bioactive compounds. And when you talk about the microorganisms, especially the bacteria and the fungi from these plants, are going to be the alternative. Since the plant tissue where these endophytes exist is an eukaryotic system, it will appear that the secondary metabolites produced by these endophytes may have reduced cell toxicity, and otherwise the plant in which they are, these plants will die. So that is the main reason why we opt for these endophytic fungi. And the host itself has naturally served as a selection system for microbes having bioactive molecules with reduced toxicity with towards the higher organisms. And not only this, extraction from the natural source has some disadvantages, right? Some plants depend upon the seasonal uh, climatic conditions, geographical regions, ecological problems involved in extractions, and thus require an innovative approaches. So one can ask me whether these endophytes are present only in medicinal plants. No, these endophytes are present almost in all the plants, including seaweeds, sea grasses, and other medicinal plants, including medicinal plants. So there are reports of microbial endophytes which mimic the bioactive compounds as produced by the plant itself, thus making them a promising source of novel molecules. This is the thing which makes these endophytes very unique. Whatever the plant produces is being transmitted, the character is being transmitted to these endophytes, whether it is a bacteria or the fungi which resides in the plant, which harbors these characteristics. So this becomes a very important one, to harbor these endophytic fungi for our human use, because these fungi can be grown in the lab. They can be produced in huge amount by using fermentation techniques. And apart from this, the the cost that is involved in the production of the drug is also reduced. And mostly people may disagree with me, because when you talk about these fungi, you talk about the fungi with spores. But when these are transferred to the industrial production, the spores are being silenced by mutation, and the fungi can be used for the production of the novel bioactive compounds. Right. When you talk about the endophytic fungi, there are certain criteria that are used for selection and identification of the plant. Plants from distinct ecological environmental niche, growing in unusual habitats, holds a novel strategy for the survival should be considered. And plants with ethnobotanical origin can be selected. And plants that are having widespread present in the region, having unusual longevity, or have occupied a certain prehistoric landmass, are selected for the study. And when you talk about these things, the plants which are occupying unique region, or plants inhabiting harsh environments or extreme environments, like the mangroves. In Tamil, we call it as Sadupinila Kadagal. Even the mangrove plants host certain bacteria and fungi which are having potential bioactive compounds that can be utilized against these uh, microorganisms which are supposed to be resistant against several drugs. So when you talk about the isolation of the endophytic plant, the first foremost thing we should do is the surface sterilization of the plant, because any plant tissue will contain epiphytic bacteria. So to remove these epiphytic bacteria, the first and the foremost thing is surface sterilization of the plant tissue. This is done usually by sodium hypochlorite and followed by 70% ethanol. The reason for using this is sodium hypochlorite and ethanol is very sublimate, and it will not destroy the plant part when compared to mercury chloride, which is also used in plant tissue culture for surface sterilization. So when you talk about surface sterilization, depending upon the nature of the tissue, the timing of the sterilants varies. If it is a leaf or a flower or a root or a stem or a fruit, depending upon the intensity of the plant tissue. If the plant tissue is going to be very hard, you are going to subject it for more timing in order to remove the epiphytes that are attached to the plant tissue. Right. When you talk about the method of isolation, when the plant part is surface sterilized, there are different methods of isolation of endophytes. Certain people follow what is called as, they grind the tissue and they try to isolate it by plating it on a media like sabrodextrose agar or a potato dextrose agar. But normally what can be done is, they can either grind the tissue, or They normally what they do is they cut the portion, do a surface sterilization, and they place the tissue of the plant in the agar. And this method we call it as agar impingement method. Along with the leaf, stem, root, which is being processed by surface sterilization, along with this, a plate containing unsterile tissue is also kept as a control. So we have to be very sure that the fungi what we are isolating is an endophytes and not an epiphyte. So that is the main reason why we always keep for a control plate along with the processing plant tissue sample. Once there are different medias that are being used, namely potato dextrose agar, malt extract agar, and sabro dextrose agar. So depending upon the some people go for sabrodextrose agar, but we normally prefer potato dextrose agar for the isolation, because some ingredients in sabrodextrose agar is supposed to be an inhibitory for certain fungi or certain fungi which are very slow growers. For example, for the fungi, when you isolate it from the mangrove plants, we normally go in for potato dextrose agar rather than a sabrodextrose agar. And apart from this, sabrodextrose agar is a clinical media that is used for isolating fungi from clinical samples. And normally, potato dextrose agar is the preferred one. So this is the step that is depicting the isolation of endophytic fungi from the plant. You just wash it, give several washes. That is, first put them in sodium hypochlorite for say about 50 to 60 seconds or 1 minute, depending upon the tissue. Have a 2 set of washing in sterile distilled water, followed by a dip in ethanol. And finally, you wash it in sterile distilled water and cut the cut portions is placed on the plate, and the plates are kept at 28 to 30 degrees C and observed for the growth of fungi. So this is just to give you an idea about how sampling of leaf, root, seed and stem segment of a plant called as Andrographis paniculata, which is kept on sabrode dextrose agar. So for certain medias, for certain depending upon the condition, I have used here sabrode dextrose agar. Here you can see the plant tissue that has been and the fungi that has been grown. So very beautiful growth of fungi. And apart from this, when the growth comes, normally we make a wet mount. We call it as by using a lactophenol cotton blue staining, we identify the fungi. When you go for the identification of fungi, normally the fungi can be isolated. Once it is been isolated, once it is grown, the fungi can be subcultured on a media called as potato dextrose agar. But when you talk about certain species of Aspergillus, like Aspergillus flavus, fumigatus, and niger, we use a media called as Zepecdox agar. This media is used for differentiation of Aspergillus species, and it gives a distinct pigment that can be observed on the front side of the plate. So here in this picture, you can see this colony appearance, the front and the reverse side. It is very important that when you work in fungi, for certain dermatitious fungi which produces pigments, the reverse side is also very important. So this picture shows Aspergillus flavus growth on the front and the reverse side. And here on the right side, you can see the microscopic appearance. So you can see the gonadial head where you can see the sterigmata appearing. So this is a photographic image of Aspergillus niger that is grown on Zepecdox agar along with the microscopic appearances. Here you have an organism called as Curvularia. This is a dermatitious fungi. And here you can see a very distinct pattern on the left side where you can see the front as well as the reverse side. When you see the reverse side, you can see the pigments that are being present. And this is the characteristic feature why we always, when you talk about endophytes or any fungi, it is better and it is preferable that you see the front as well as the reverse side of the plate for pigmentation for these dermatitious fungi. On the right side, you can see the microscopic appearance where I think the picture is very little. You can see the spores that are being distributed in the microscopic field. And this is the appearance of Aspergillus nidulans, and right side is the microscopic appearance of the fungi. Right. When you talk about the identification, the conventional method called as the microscopy following lactophenol cotton blue staining is usually the standard method. But certain fungi which do not sporulate, these fungi are made to subculture on sporulating media like potato dextrose agar or corn meal agar or tap water agar. The main idea of making subculturing on these media is you are giving the fungi a stress condition where whenever, in a basic microbiology we have studied, whenever an organism encounters a stress condition, it turns in the form of spores. So with that idea, we make the fungi to make a subculture on these media, and we will identify the fungi. So this image shows the macroscopic and microscopic features of isolated endophytic fungi. And here I have shown you very clearly the dermatitious fungi and along with the aspergillus. When you go into the literature, there are about 18 to 20 different species of aspergillus, but ours, for we have studied only namely flavus, fumigatus, and niger. So you have Aspergillus terris and you also have Aspergillus clavatus. So for all these things, we require what a media called as a Zepactox agar and an identification manual by which we are able to identify these fungi. So for certain fungi which even do not sporulate on these media, they are taken for the molecular characterization. So molecular characterization is usually done by isolation of the DNA and amplification by using ITS region of the fungi, which is a major step that is used for the identification of the endophytic fungi. So when you talk about when you grow these fungi, when you make a subculture, the second idea is production of or extraction of the bioactive compounds. So when these fungi live in symbiotic relationship with the plant, they are able to acquire the characteristic of producing bioactive compounds. So these fungi has to be subcultured on a liquid media. There are two ways of doing it. One is a solid state fermentation or a submerged state fermentation. Solid state fermentation is the main idea of subculturing these fungi on these liquid media is to extract the bioactive compounds. So solid state fermentation is widely used for bioactive compound production from fungal endophytes. And these biomolecules are mostly metabolites generated by the endophytic fungi when the fungi is made to grow on solid support selected for this purpose. So it can include any variety of solid substrates such as wheat bran, rice bran, coconut oil cake, or a vegetable waste, or anything that can be used with pure culture of endophytic fungi. Sometimes, because many people may ask why this solid state fermentation, some of these fungi they breed on decomposing vegetable combinations in a natural environment. That is why we are giving under a solid state fermentation. But normally, what we use in our laboratory is a submerged fermentation, right? To extract the bioactive compounds or any enzymes from a liquid broth culture. And normally we use a PDB broth for isolating the bioactive components from the endophytic fungi. The broth is prepared and 5 days old culture is taken and it is put into the broth and it is kept under the shaking conditions. And this is the picture. It is kept in a shaker and for 10 to 15 days, depending upon the bioactive compounds that is being looked for. Later, the broth is taken and the broth is filtered and the mycelium is separated. I will come to it. And this is the picture shows the solid state fermentation and a submerged fermentation. And this is when you do this, and using the crude culture, we check the activity of the fungi. This is just an initial step. And usually when depending upon the conditions, for example, we check the activity by using standard stains. And if it shows any activity, we will take it for the screening procedure. But normally, before doing this procedure, we grow these fungi on a plate. We do a primary screening, and those fungi which shows activity against the clinical strains by primary screening by agar block method will be taken for this submerged fermentation. So when you talk about the media that is being used, a lot of things are underway. The media optimization of the media conditions like carbon, nitrogen source, what is the nature of inoculum, what is the incubation period, what is the temperature, and what is the pH. And for these, depending upon the bioactive compounds, what you are going to use, or what you are going to go for, we need to optimize these conditions before we go for the secondary screening. And these are the examples of the media used for the production of bioactive compounds. An example of certain endophytic fungal medias, which are now used in currently in the research. And this is a condition where it is taken in it is kept in a shaker under organized condition. And some bioactive compounds require organisms in a static condition also, where the endophytic fungi is inoculated and kept in the lab without any kept in a lab. It is left undisturbed for a few minutes, for a few days. So once these compounds are taken, it is extracted. It is separated and it is extracted by using any solvent, where the two phases get separated and the topmost phase is taken and it is evaporated and the extract is prepared. This is how it is done. And this is, you can see the left side of the image shows the endophytic fungal which has been prepared by using the uh, extractor. And here the topmost phase is being taken and it is kept in a evaporator and for a few days, and finally the extract is taken, and this we call it as a crude extract. Normally we check the activity of this crude extract, and we need to, once it shows the activity, the fungi has to be cultured on a large scale to get a larger amount of the extract. And this is just for an initial study which can be done. But when you go for a larger volumes, it requires larger volumes of the media as well as the solvents that are involved. So once we have extracted this compound, this we'll dissolve the crude extract in any solvent, and it is subjected to what is called as the GCMS analysis to find what are the compounds that are being present. But normally, when you talk about the purification, we need to grow these fungi, isolate the bioactive compounds by this method, and run a column to know which fraction is able to produce the bioactive compound. And we need to go in for a further purification step to actually crystallize the nature of the compound. And for this, we need lot of instruments, and mainly we go for chromatography followed by NMR and mass spectroscopy, and to find out the nature of the compounds. So when you talk about the solvents that are used in this process, depending upon the compound of your interest, the solvent also varies. It is not just like that we can use any solvent for any compound. You have to go by the literature. You have to study a lot of reprints and find it out who are the pioneers or who would have worked in all these aspects in your compound previously to know which solvent is which necessary for the extraction process. Otherwise, it is going to be a big task for you to choose a solvent for extraction of the bioactive compound from the endophytic fungi. Just to give you a glimpse of the anti-cancer compounds so far that has been reported from the endophytic fungi, and the king of this is the Taxol, camphothecin, vincistin, and vinblastine that have been isolated from Taxus brevifolia, Camphothecia accumulata, and vincistin and vinblastine, which have been reported from an endophytes alternaria species that have been isolated from Catharanthus roseus. We call it as Nitya Kalyani or Sudugatumali in Tamil. So these are the research that have been going on currently in the field of endophytic fungi. And apart from this, these endophytes are also present in mangroves. We call them as Sadupu Nilakadagal. So this area is inhabited with high salinity, and these fungi are present in a highly stressful conditions. So this makes them very unique and novel to produce some bioactive compounds. And here also, unlike like medicinal plants, these mangroves also produces bioactive compounds. These endophytes also produce bioactive compounds similar to the plant which produces. So those plants which produces any bioactive compounds, the endophytes isolated from these plants is also said to mimic the same characteristic of the host plant. And now, apart from this, seaweeds uh, is also reported. There are a lot of works with regard to the seaweeds, which also harbor these endophytic fungi that has been produced, and they are found to produce several bioactive metabolites and which are having lot of activity. And these have been reported in the literature, and this research is mainly going on. And we are also working in some of the seaweeds and the medicinal plants in our laboratory in Periyar University, where we are in the process. And one such compound we are trying to find it out is the camphothecin, and 2 of our students are working in this aspect. And this is with regard to the endophytic fungi. And so I would like to thank the organizers for giving me an opportunity. I would like to thank the organizers for giving such a wonderful opportunity for making my presentation in this prestigious national webinar. I would like to thank the organizers, the principal and the chairman, and the organizing team of microbiology department for this wonderful opportunity. Thank you.
Thank you, sir. We have few questions from the audience, sir.
Yes, ma'am.
Can parasitic fungi evolve to control humans?
Parasitic fungi
Evolve
Repeat.
to control humans.
Can parasitic fungi evolve to control humans? You
Yes,
mean
sir.
to control human diseases?
To control human diseases. Yes, sir. Yes, sir.
This depends upon the nature of the fungi, ma'am, because we can't say that all the fungi are not villains. But at the same time, if they are not villain also, we can't say the nature of the fungi, because you say it is a parasitic fungi. So we need to determine the virulence nature. If they are not virulent, you can use it for. But there are a lot of issues that are related with it.
Okay, sir. And another question, sir. Which part of the plant is the best source for endophytic isolation, and does it differ with the nature of the plants?
Yes, certainly it differs with the nature of the plant, because when you talk about any endophytic fungi, whenever you go for selecting the plant, we first go into the literature and see which part of the fungi is able to produce the bioactive compounds. For example, if the leaf is able to produce some compound which is very effective against some dermatophytic infections or any infection, we normally concentrate on the leaf part. But depending upon the literature, only we can say. We screen them based on the literature.
Okay. And one more question, sir. Can cordyceps fungi be a promising agents of biopesticides and biocontrol agents? The so-called
Yes,
zombie fungi.
zombie fungi.
Yes, sir.
We don't know, ma'am, because the nature of the microorganisms cannot be predicted. Because one simple example is the COVID virus. As per my prediction, we are going to have two more viruses, which are going to come from the animal state. So this our favorite virus is not going to be the last. We are going to have still 3 more fungi which are going to be coming from the animal side. From the animal, it is going to be transmitted. and we are going to face that new trend. With regard to the this viruses or the zombie virus or whichever it is, any virus that can be cultured in a laboratory and kept as a safe, it is good. Unless and until when man tries to mingle with the virus and try to make it disturb the virus and disturb its nature by doing any mutation, it is going to show its nature. Similarly, the fungi also.
Yeah, nature has its own way.
Yes, ma'am,
And
definitely.
yes,
And we have disturbed the nature, and we got this COVID virus. The virus that was grown in a laboratory has been transmitted to the other region. So certainly, people predict that the coming viruses, two more viruses are going to come from animal origin because man has started eating all sorts of meat without doing any proper processing.
okay, sir. And one last question, sir. Can you suggest few
Yes.
endophytic fungi to identify antivibriosis activity?
We can't sell, we can't say, or we can't just predict like there are certain endophytic fungi we can screen antivibriosis. Those plants which are used, the bioactive compounds that are active or can be used against vibrio can be a good source of uh, for this. But we need to screen those plants for the endophytic fungi. Screen the endophytic fungi, take the endophytic fungi and check for the activity.
Okay, sir.
That's how it can be done. We can't blindly say that these endophytes have this activity unless and until we screen them.
Okay, sir. Yes, Maha, you can take over.
Yes. I hope the questions have ended.
Yes,
Is the
Maha.
question and answer session over, ma'am?
Yes, yes, it's over. It's over.
Okay.
Yes, ma'am, it's over. Yeah.
So, thank you so much. So, so fungi are more beneficial to mankind and the environment as compared to the diseases they cause that are opportunistic. So it has become evident from your session, sir. Thank you very much for being with us. Before we go on to the vote of
Yeah,
thanks,
thank you,
I
ma'am.
request the participants. Thank you, sir. I request the participants to kindly fill up the feedback forms. The link would be displayed in the live chat now. The certificates of participation for our webinar would be mailed to the participants who have filled up the feedback forms within a week. I request Dr. Lakshmi Priya, Head of the Department of the PG Department of Microbiology, Dr. MJR Janaki College of Arts and Science, to deliver the vote of thanks.
Good afternoon to one and all present in this platform. On behalf of the management, principal, faculty, and students of Dr. MJR Janaki College, I am glad to be here to express my vote of thanks. My heartfelt gratitude to our chief guest, Dr. Thangam Menon, Professor Emeritus, Department of Microbiology, Savitha Dental College and Hospital, Chennai, for accepting our invitation and enlightening us on how COVID-19 increases the risk of fungal infections because of its effect on the immune system and also due to the treatment with steroids and other drugs that weaken the body's defense against fungi. I'm thankful to Dr. Bernard Rodriguez, Senior Professor, Botany Discipline, School of Biology and Biotechnology, Goa University, Goa. His presentation and explanation about mycorrhizal fungi was simply brilliant. Thank you, sir, for elaborating on how these obligate root biotrophs exchange mutual benefits with the plant families. My thanks are due to Dr. Prabhu, Assistant Professor and Head in Charge, Department of Microbiology, IBMS, University of Madras, Chennai. He spoke on opportunistic fungal infections and how standard precautions are designed to both protect and prevent from spreading infections. The final speaker of the day was Dr. Aravind Prashant, Associate Professor, Department of Microbiology, School of Biosciences, Periyar University, Salem. Thank you, sir, for detailing on how endophytes establish relationship with the plant to border on pathogens and on their biocontrol mechanism. Thank you, speakers, for all your valuable time. Teamwork works well. I thank my team of faculty members for their support. A word of appreciation to Dr. Nitya Kalyani, Head, UG Department of Microbiology, Ms. Mahalakshmi and Ms. Preeta for their technical assistance. My gratitude to our correspondent, Dr. Lata Rajendran, our chairman, Dr. Kumar Rajendran, our principal, Dr. Mani Megalai, vice principal, Dr. Lakshmi Balaji, Dean Academics, Dr. Shanti Lakshmi, and Dean Students, Dr. Abhida Sabapati, for their constant encouragement. Finally, thank you, dear students, for all your active participation. Thank you, one and all. I request the team of faculty members to kindly freeze for the photo.
Bernard sir and Arvind sir, you can also join. Arvind sir. Arvind sir. Thank you, Lakshmi ma'am.
Kindly raise for the national anthem.